Hold on. Sophia, don't come back in that door though. Really? Yeah, let's wait. We said 1:05, so well, I mean we do a little welcome, but.
All right, just a reminder, everyone. I'm going to be sitting right there behind Matteo. We have 2 minutes left. So at 13 minutes, I'm going to at 15 minutes, I'm going to give you a 0. And at 19 minutes, meaning like you've already gone mostly through your entire question time, I'm going to get up and start. Okay? So if I'm standing over here, like, know that you just need to, like, wrap up. You need to wrap it up pretty quick, and we'll probably just take one, like, quick question when we're transitioning to Q&A. Okay? Question? Use the up and down arrows or the clicker to advance. You probably will. It's hard to see. I mean, as some of you experience, it's hard to see your mouse cursor here. So if you need to point something out, you probably just want to point it out. Otherwise, it'll be like lost.
Everyone good? Just do a little shake. Stand up. It really works. Like it does work.
Oh, it's kind of
It's like for real.
Felicia, we are wonderful kind of illustrators. You'll be across lunch shortly. Okay. Everybody set?
Everybody set? All right, here we go.
It's gonna be amazing.
You're gonna do great. All right, hi everyone. Welcome back to the second half of our final presentations for the SSI 2024. If you were lucky enough to join us this morning, you saw 5 awesome presentations, and you have a lot more in store for you this afternoon. So we have a lot of great talks to hear. So without further ado, I'm going to welcome Julianna Escobedo, who joins us from Wellesley College, and this summer she worked with Shannon Tushingham. Welcome, Julianna.
Thank you, Rebecca. So I'd like to start with an introduction in my native language. So, Hakuhilpo, Julianna Nituanea, Akikitamneo. Hello, everyone. My name is Julianna, and I'm Kintanamak. So this summer, I'm a part of the anthropology project under the guidance of Dr. Shannon Tushingham. And my project is titled Platforming Native Communities to Steward Cultural Heritage, Two-Eyed Seeing at the California Academy of Sciences. So you might be wondering, what is two-eyed seeing? So it's important to apply this concept to museum practices, and it's basically a concept where Indigenous knowledge holders and scientists engage in mutual learning and collaboration. And so this approach can really provide a lot of insights that maintain community autonomy and preserve and protect cultural heritage and ecological heritage for Native groups. And so we kind of summarized two-eyed seeing into 3 principles. So that's reframing expertise. And so this is about acknowledging that both Indigenous knowledge holders and scientists are experts. And the goal is really to recognize their unique expertise and encourage collaboration. And so the second principle is building relationships. And so it's important for researchers to create long-lasting relationships and to build trust with local Native and Indigenous communities, and really emphasize that science isn't about them or for our own gain, but it's for them and for everyone's benefit. And so the third principle was to listen and apply. And so we really needed to listen to folks we were having conversations with and apply their suggestions to museum praxis. And so museums like CAS can incorporate these perspectives on objects or in research projects, and it really encourages institutions to present both perspectives alongside each other, making every story more whole. And so this is a photo of me and Chantel in the collections room. And so I had a lot of big research questions, and, you know, coming to CAS and being in the museum, I really recognized that museums are and have been repositories of colonial history, and sometimes they can silence or misrepresent Native Americans and their artifacts. And this can really just obscure or diminish the traditional knowledge held by each Native creator and embodied by each object. And I will say that the new California exhibit in CAS really does not do this at all, and it was really well made, and I love walking through it. So thank you to the exhibits team. And so my research questions were, how can we transform museum spaces and listen to Native peoples and the story they want to tell? And really, what do museums owe to tribes whose artifacts that they possess? And so where did we begin? We started by making these really cool flyers. So my kind of partner in crime this summer was Chantel, and she did all the artwork for these posters. And so we had the Snapshot Cal Coast event that we wanted to invite folks out to, and Chantel knows that abalone is my favorite, and so she drew these abalone, and I kind of really just did the languaging for these posters, and I really just wanted to emphasize that Native people can have a good relationship with museums, and by coming into our collections, it can really contribute to their revitalization and sovereignty efforts, whether that be like language or art or really whatever. And we also thought it was really important to go to them and really go to these Indigenous-led spaces and just kind of have conversations and come with physical materials that they could carry with them. And, you know, these posters, so these posters wouldn't really be lost in like a email inbox, you know? So, yeah. So early on in my internship, me and Chantel got to go to the Gorman Museum in Davis, California. And this museum is completely dedicated to the display of contemporary Native American artworks. And I got to meet with the curators, Victoria and Hulea. And I discovered that the Gorman has actually worked with CAS before, and we have paintings in our collections. And as someone who studies art history, that was really interesting to me. And so I kind of was really pressuring my mentor and Laura, the collections manager, to kind of let me go back there. And I had never been to a collections room before. So you got to see like the typical pots and baskets and jewelry and textiles. And it was really just inspiring and moving to be in those rooms and with those objects. So kind of seeing those paintings though left me with a lot of questions, like how were they collected? When did they get to CAS? You know, what stories do they tell? And ultimately, is it really fitting for these paintings to be at a science institution? And, you know, have these paintings ever been on display? Will they be on display? And, you know, I think a part of me felt really obligated to kind of bring these items out from the darkness of the collections room. And so you'll kind of notice that these paintings are really, really unique. And you can see this floating. They're cultural depictions and ceremonial depictions. But there are these figures that are kind of floating in space. And that's pretty atypical for art to, you know, not have a background. And so, you know, I discovered that these paintings were all made from around 1920 to 1930 under the Santa Fe Indian Boarding School art department. And it's really just painful and violent time period. You know, these kids are being told, you know, to paint their traditional, ceremonies, but at the same time, their hair is being cut and their language is being lost. And so it's these paintings are really, really interesting and carry a lot of history. And so I mentioned earlier that I met the curator at the Davis Museum, and this is Hulea. And doing research on the paintings I actually discovered that we have paintings that were made by her father, who was also an artist. And so she, I kind of talked with my mentor, and I really wanted something kind of tangible out of this experience. And so we decided to write an article for a Native news outlet, just talking about all the research I've done on the paintings. And Hulea actually agreed to interview me, which is really exciting. And so we're just kind of talking about big ideas, like, land loss and the environment and their connections to their descendant communities. And so we realized that these objects are living beings and they remain inextricably tied to their descendant families and communities. And so this concept really applies to all the objects that we have in our collections. And so we really just want to have conversations around, a shared stewardship plan in which descendants really have a say over how these items are cared for and displayed and, treated. And so and so we just think that with their rich and cultural historical significance, they deserve to be seen and appreciated. And so, that leads me into the second half of my internship. And so we took a trip to Berkeley, and we went to the 5th Annual California Native Ways Festival, and it's put on by the California Institute for Community Arts and Nature. And there we had the pleasure of meeting Julia and Lucy Parker. You can see them. This is them today, and this is them nearly 60 years ago doing a demonstration at UC Berkeley. And they were just really awesome, and they sing songs and were really excited to talk to us, which was really nice, kind of like my first time going out in the field and having conversations and people being enthusiastic about talking with me. It was really, encouraging. And so, they mentioned that when they came to CAS, they mentioned that they had a relationship with the Institute, and they used to come under the traditional arts program, and they used to perform and share their knowledge about basketry. And they expressed really a desire to come back, and they were kind of like, oh, what happened to that? And so me and Shannon were writing notes, and we were on it like, what is this program that they were talking about, and really how can we bring it back? And so we had these big questions like, how did it begin? Why did it end? What's kind of the merit of bringing it back? And so we discovered that the traditional arts program ran from 1983 to 2006. Its objective was really just to broaden the Academy's engagement with the local community by giving artists the opportunity to participate in a diverse spectrum of, cultural heritage. So this included music demonstrations, dance demonstrations, food demonstrations, and just craft making. And it was just all kinds of expressions of daily and ceremonial life for a bunch of different cultural groups. And so, this picture up here is a photo of Phyllis Wattis. She is the benefactor of this program, and she actually established an endowment to a $1 million endowment for this program to exist and to run. And so, we had, but the big question was, where is the money? And, where does it still exist? And we actually found out that this endowment does still exist. And so Shannon and I are doing some investigative work about how can we get this money back to the anthropology department, and how can we revive this program. And so ultimately our big question was like, what's the merit of bringing this program back? And so, the mission of the California Academy of Sciences is to regenerate the natural world through science learning and collaboration. And so by bringing in artists from diverse backgrounds, it'll help make the museum more inviting and welcoming. And, it'll create an engaging environment that sparks curiosity and fosters creativity. And, you can build connections with the community. And so ultimately I was curious about what CAS's responsibility is to, present and preserve cultural narratives, particularly those of Indigenous and underrepresented backgrounds. And ultimately they should do this in the best, most accurate, and most contemporary manner. And they can do this by reviving this program. And so this whole summer, I've learned that systematics traditionally relies heavily on quantitative data like morphological features or genetic analysis. However, anthropology and qualitative research that I've been doing all summer can really offer valuable insights that complement this approach. And so anthropology really just helps us consider the cultural and social factors that might influence how communities interact with and classify the natural world and, museums. And so talking directly with people was really essential to my summer. And I'd really like just thank Shannon, my mentor, for emphasizing the importance of going out in the field, but in a different way, and, building relationships and documenting Indigenous knowledge, and, really for nurturing my interests in art and culture, and really helping me imagine a future where this work becomes fully realized and supported in all institutions possible. And so, thank you to everyone.
All right. Does anyone have any questions for Julianna? Nice.
I had a question. Being in research and understanding the differences that you've seen in collections while you've done your time this summer, how do you feel as a Native woman that you're going to make changes and you're going to implement them outside of this program? Are you interested in policy? And then how are you going to bring in all tribes?
Yeah. So, I guess this is like the first museum I've worked in and the first space where I've been able to work with collections. And I think I'm really inspired, to go out and work in other museums and really, just kind of further this research and work I've done. I think I'll carry this with me for the rest of my life. And that really is my, career goal, is to just go out and kind of infiltrate museum spaces and really do it in this manner. So what was the second part of your question?
Just how to incorporate what changes you want to make to include all tribes, because there's so many different collections that need to be addressed and that haven't been for tribal people of all areas.
Totally. And just coming as a Southern California tribal member, a member of Tohono Indian tribe, and whose some artifacts we've had in CAS, I go to college out in Boston. And so just kind of doing cross-cultural examinations, I really want to get away from the pan-Indianism and, really recognize that each tribe is very unique. And I think it's really, I hope to do that in my work. And I think going to school out on the East Coast and being from California has been really helpful, to do that naturally. Yeah.
That was so awesome. It was so wonderful to work with you and Chantel this summer. That was really great.
Yeah, thank you.
And I'm really curious, kind of building off of your questions, if you have ideas about sort of, how we might do a better job. We've talked a lot about blending Native science and Western science and through collections and this kind of work. Is there anything that you think, if you had to cherry pick one project, is there something that really you'd go for or prioritize?
It's hard to say. So me and Shannon went to Oregon last weekend to fish camp, and I think I really like going out in the field and, kind of just meeting people. And gosh, I don't know. I think there's a lot of merit in both projects. I think a part of me is I love art. I study art history. And so I think, kind of going back to your question, I think, I think kind of taking on a more contemporary lens. So whether it be through contemporary Native arts that people are making is really important to me. But I don't know. I think this programming the traditional arts program, connecting it in some way with science. I don't know. That's kind of hard to imagine, but I think if I had maybe a little bit more time, I guess I could have got more into the kind of ecological side of things for sure. Yeah.
Awesome. Thank you so much,
Thank
Juliana.
you.
Just one super quick second. We just have to do a little re-uploading, and then we'll resume our scheduled presentation. If only I could see the mouse cursor. Okay, and we're back. Next up, and quite honestly down to the wire, Matteo worked through the night up until 5 minutes ago to get all of his data back and his presentation together. We have Matteo Alemany, who's joining us from Willamette University, and he worked this summer with a team including Maddy Hannappel, Anna Holmquist, Athena Lam, and myself.
All right, I'm Matteo. I worked with Lauren and Maddy this summer on orb weavers of the genus Tetragnatha. And this is a genus of spider with global distribution. It's a very abundant genus. There are over 300 species recorded, and it has a worldwide distribution. It is found on every continent in various riparian ecosystems. A big part of why this species is important to study is that it is important in studying the mercury cycle and how mercury pollution affects aquatic ecosystems. Because it preys on primarily like emergent aquatic insect prey, it's a great way to study how mercury contamination moves from aquatic ecosystems into terrestrial ecosystems and how mercury magnifies as it moves up the food chain. So that is what Maddy has been studying with her research, and that is the project that I joined at the beginning of the summer. A great reason like why like they're a good indicator is that they're very easy to collect. So this is what the global distribution of Tetragnatha species looks like. This is from Bold. As you can see, there are lots of different recorded occurrences in North America and in Europe. A difficulty with Tetragnatha is it's very difficult to identify them to species level. They are extremely similar morphologically. So individuals of different species often have identical color patterns and sizes, while individuals within species may have like wildly divergent color patterns and sizes. The only reliable way to identify them is to study their genitalia, which is only really possible with males. To study adult females, you have to dissect them, which is not possible if you're doing testing like mercury testing, which requires you to preserve the full body of the specimen. And you can't identify immatures using the genitalia at all. So the goal of this project was to try to find a way to identify these spiders to species level without having to either destroy them physically or like study their genitalia, which is a very difficult thing to do. This example of like one of their male's palps. The last major revision of Tetragnatha in North America was published in 1981. That was using almost entirely morphological data. So while it is useful for determining like what males correspond to which species, it is not very useful for trying to identify females or immatures. And it's not based on any genomic or genetic data at all. So, yeah. These are the 7 species of Tetragnatha that are native to California. Each of them has a different palp structure that you can use to ID the males of the species. The ones that matter most to this presentation are going to be versicolor, laboriosa, elongata, and shoshone. Those are the ones that we found most abundantly collecting in California. These are their distributions. As you can see, like guatemalensis, nitens, pelescens, those are very infrequently found in California, while the others are much more frequently found and much more widely distributed across the state. This is versicolor. It is by far the most abundant of any of the species that we're going to be talking about here today. It has a distribution across North America, and almost all of the spiders that we collected at each of these sites ended up being Tetragnatha versicolor. This is elongata. This is shoshone. And this is laboriosa. So the objectives of this project were to use DNA barcoding to help identify Tetragnatha spiders, and then to compare that DNA-based identification to morphological characteristics, with the eventual goal of making a morphological key for identification. So spiders were collected from 9 sites across Central California, and then we measured leg length and carapace width, and then also collected and photographed palps in cases of the males. We had hundreds of spiders that we could have used for DNA barcoding. We ended up attempting 108 samples. We extracted DNA from them using the QIAGEN DNase blood and tissue kit. And then our original plan was to use LCO and HCO primers to amplify the CO1 barcode region. That did not end up working out. So we ended up trying a total of 6 different primers to try and amplify the CO1 gene. As you can see, this is a compilation of all of the various failed gels for each of the different primer sets. I think we tried LEP-F1, LEP-R1, we tried degenerate lepidoptera primers, we tried mini barcodes. It all consistently failed for no clear reason, which is super strange because this should not be a difficult genus to barcode. So we don't know what that's about, but it's very interesting. But we were able to get DNA back finally. We so far have only had success using the Lepidoptera primers. That's that on the left over there. And using FWH mini barcode primers. That is this, the gel on the right, the top row. So far we have 30 successful barcodes of our 108 samples. We have more that we still need to clean and then try to, identify using GenBank and bold sequences. But of those, the overwhelming majority of them are Tetragnatha versicolor. A small remainder of them are laboriosa, and then only 2 of them are Tetragnatha elongata, or were identified as Tetragnatha elongata on GenBank. However, they were only 94% similar to any of the sequences on GenBank, which is that's on the lower threshold for species level identification using sequence divergence. So it's unlikely that these mystery specimens are actually members of Tetragnatha elongata. We don't know what they are yet, but hopefully we'll be able to find out. So in an effort to try to identify, like figure out what they are, we took samples from the museum's collection. These are like archival specimens that are over 90 years old. We tried to get one for each of the major Tetragnatha species in California, including Elongata, laboriosa, versicolor, and Shoshone. And our plan was to extract and amplify DNA from these and then use it as like a reference to determine what is actually like a member of Elongata versus a member of versicolor versus like a member of Shoshone. And we were able to successfully extract and amplify that DNA, which is very surprising. It worked even better than the fresh DNA that we had collected from the other Tetragnatha. We're unsure why. So far, we have gotten one sequence back, and it is a 100% match to all of the Elongata sequences on Barcode of Life database and on GenBank, which is good. This is a maximum likelihood analysis created with all of our samples. We can see that like laboriosa forms a nice monophyletic grouping, as does our Elongata. The samples on the top, the two 182 and 188, are the mystery ones that were only 90% similar, but they're more closely related to the other Elongata, like the archival specimen, than anything else. So maybe they're actually Elongata. We don't know yet. Versicolor looks a lot worse. You can see here that like there are some Tetragnatha Shoshone within this grouping. That either means that like this is a species complex and that like Tetragnatha and Shoshone are like not actually like separate species, or it means that people are just not correctly identifying these specimens when they're uploading them to GenBank and Bold, which is totally possible. For example, like that laboriosa specimen down there is almost definitely a symbol of misidentification. So it's not clear whether it's possible to use this data as a way to like reliably identify like species, because we don't know that any of the species on GenBank or in Bold have been reliably identified by experts. We compared the morphological data we collected across species. It doesn't seem like there are that many trends. Elongata tends to be larger in leg and carapace length, but there's significant overlap between like versicolor and Shoshone, which means that it's not necessarily a useful tool for distinguishing those species. So we have even more DNA that we are hoping to sequence from those specimens, and hopefully they'll be able to give us a clearer picture on morphological trends across species, if there are any, as well as the archival sequences. Hopefully we'll be able to use those to try to figure out the mysteries regarding like the taxonomy of Tetragnatha, versicolor, Shoshone, and Elongata. The eventual goal of this project is to create a morphological key that we can use to reliably identify these species using morphological data. And that's it. Thank you, Lauren and Maddy, for your support during this project, as well as Ed, Rebecca, and Luigi. You guys have been really great at making SSI feel like a super welcoming environment. I'd also like to thank the rest of the Archaeology Lab, Athena Lam and Grace Kim in the CCG, and then Anna Holmquist and the insect barcoding team. They're the only reason like we were able to get any data at all. So like, thank you guys so much for your help. And also thank you to the rest of the SSI interns.
All right. Awesome work, Matteo. Any questions?
was a great talk. Congrats on getting some data. So that last morphological analysis that you showed, how are you grouping the individuals into species? Was that based on the barcodes?
Oh yeah, this is based on barcode IDs. So like all of this is like what it came back as like on GenBank or on Barcode of Life. That's what those colors mean. Yeah.
Here, I have a quick question. You addressed it a little bit, Matteo, but you know, it looks like there's a real problem with knowing if people ID'd things correctly on GenBank. And this is actually something that all of you dealt with, right? Like blasting against GenBank and then figuring out where your specimen fits in. Like, what do you think are some ways to help get around that? Like you proposed some ways.
Sure.
But
what really surprised me is that like the specimens from Barcode of Life, which to my understanding has much stricter submission like criteria, were also not reliably identified. Like all of these ones in italics are stuff from Barcode of Life, not from GenBank. So like in theory, they should have been reliably identified by experts to the species level, if they're up there and they're public with that level of identification. But that doesn't explain why we have like a Tetragnatha Shoshone just like sitting in the middle of our family of versicolor. And that's just like the two specimens that I pulled. Like I guarantee that if I had pulled even more, that like it would not look like there are two separate families. Like it would just like look like they're all like the same like species group. I believe that there's actually was a paper study that like showed that like most of the versicolor Shoshone might have been mis-ID'd on GenBank and on Bold, which is annoying.
yeah, it sounds like the key that you're thinking about making for like community folks to use is going to be really useful for everyone to use, right? Like if you assume those experts were using a key, like there's some issues with how things are keyed out.
Yeah.
Oh, there's one more.
Hi, Matteo. That was great. And I'm happy you got some bands for your recent gels. I'm just wondering, is this different genes? I was just seeing potentially there was one that I saw that said 18S versus, I'm just curious what the data was for this tree.
This is all like CO1.
Okay.
They're different primers. So like some of them like were collected using Lepidoptera F1. And then others we got using the FWH primers, but those both target CO1. So they should be showing us like similar things.
Great. Yeah. And I had one more question about the archival samples. I know you got one sequence back. Did you get any more? Is that do you think that's gonna help you define these a little bit better?
Yeah. The one that I'm really like want to get back is the Shoshone, because depending on where that lies in this phylogeny, that's going to tell us a lot about like whether Shoshone is actually a species or not, and whether it's distinct from versicolor. The archival system that we have is a paratype, so it's basically as close as we're going to get to like what the authoritative like definition of the species is. And if it turns out like that paratype isn't distinct from like any of these versicolor, then that doesn't really suggest that like Shoshone is a separate species. But we don't know until we get that data. So
You do have DNA.
we do still have the DNA. Yeah, it's just refuses to amplify for some reason. And then when it amplifies, it refuses to sequence.
So given the troubles with amplifying the CO1 fragment, do you think that there would be any use of designing custom primers from the mitochondrial genomes that are on NCBI?
That's a little bit out of my like area of expertise, unfortunately.
Those like a DNA quantity problem. Like we don't have any.
Well,
It's an extraction problem first.
no, it was a primer problem before that, though. When we did before we did the DNA plate, we had like a group of 16 samples that there was DNA in there, and yet it still like would not amplify through PCR. So I don't know what the issue is with like this genus of spiders. It is just weird.
Awesome. Well, join me in thanking Matteo. Congratulations. Oops. All right. Next up, we have Haley Arbon, who joins us from Fordham University. And Haley worked with Michael Yuan and Ed Myers. Haley.
Thank you for the introduction, Rebecca. Good afternoon, everyone. I'm Haley. I worked in the herpetology department this summer, and I'll be presenting my project out of Amazonia and back again, Phylogeography of Thecadactylus geckos. So to start, I'd like to give some context and talk about island biogeography. So this is a field that focuses on the factors that affect the biodiversity on island communities. And islands have been super important for studying evolution. A famous example is the Galapagos Islands, where Darwin studied his finches. And you might be wondering, why do islands get their own specialized field? Like, what makes islands so special? So when we compare them to the continental mainland, islands have a couple of distinct characteristics. So the first is that they're a lot smaller. So they have smaller populations, and their isolation means that populations from the mainland, there's only certain populations that are able to overcome the dispersal barrier and actually inhabit the island. So since islands have a simpler ecology, it should be relatively easier for mainland populations to colonize an island. But what about the reverse? Can island populations colonize the mainland? It's thought that this is not common or not really a thing in the field of island biogeography. And so these ideas, it's been a long-held belief in island biogeography about this idea of unidirectional colonization. So unidirectional colonization has designated continental mainlands as sources of biodiversity and islands as sinks. So once a population from the mainland colonizes an island, although that population might diverge and create new species, it's thought that these new populations cannot recolonize the mainland. However, recent studies have found more examples of this reverse colonization, so from islands to mainlands. One example are these orioles that have colonized South America from the Lesser Antillean Islands. Other examples include these short-faced bats and green anoles. Since this is a relatively recent idea, there's still a lot more evidence to be collected to fully answer this question: to what extent do islands serve as sources of biodiversity to the mainland? So my project this summer was to look closely at this question through the Thecadactylus genus. So these are geckos commonly known as turnip-tailed geckos. They have a very wide distribution throughout the neotropics, so Central and South America, as well as the Lesser Antillean Islands. And for a long time, they were assumed to be monotypic, which means that the entire genus was composed of a single species. So this is their distribution. Thecadactylus replicata was the first species and originally like the only species in the genus. It was only in 2007 when genetic analysis was performed that another species was described called Thecadactylus solomoensis, which occupies the southwestern distribution of the Amazon, in the Amazon. it took genetic analysis to discover this or describe this species, because even though it has a really wide distribution, there's very few obvious morphological differences across populations. So we have our two species, and then in 2011, there was another species named on called Thecadactylus ascroba prianorum, endemic to the island of St. Martin. And there he is. So this was not named based on genetic analysis, but only on the basis of morphological characteristics. So you can see they have this really distinct black spotted pattern down their back. So yay, we have species now, but there's still the genetic analysis that this research was based on was pretty sparse data set, and that's because, I mean, wide distribution, it's really hard to sample all of these localities. And also hasn't really been much research on the divergence of the divergence and dispersal of this genus. So how did these two mainland species diverge from each other? So considering these knowledge gaps, I had two main objectives for my project this summer. The first was to conduct the first genetic analysis on the species named on St. Martin. And the second was to examine the biogeography of the genus to try to discern their pattern of divergence and dispersal. For this objective, we looked at two hypotheses. The first follows that prevailing belief of unidirectional colonization. So for Thecadactylus, this would mean that they originated on the mainland and then diverged there and then dispersed to the surrounding areas and islands. So if this hypothesis were true, this is what our tree would look like. There would be that split of the mainland population, and then the island population would be nested within those. The second hypothesis we looked at is the boomerang colonization hypothesis. So this would be if Thecadactylus originated on the mainland and then dispersed to the islands and surrounding areas where they diverged and then came back to the mainland. And so this is what our tree would look like for that hypothesis. You can see that the island populations have diverged and that the second mainland population is nested within those island populations. So to do this, samples were collected before I got here throughout their range. And then my work started by sequencing two mitochondrial genes. So I sequenced the 16S gene from 37 samples and and the ND2 gene from 66 samples. And for our analysis, we also had samples available previously for these two genes, as well as some sequences for the cytochrome B mitochondrial gene. And then we also used a couple sequences that were available on GenBank. So we used Genius to concatenate and align all of these samples together, and then created a phylogenetic tree using BEAST with a total of 109 samples. And then for another part of the analysis, we looked at DNA polymorphisms in the ND2 gene. We chose the ND2 gene because we had an ample amount of sequences for it, and it also had better resolution than the 16S. So here is the tree. The orangish yellow represents the Solomoensis species, and then the blue and the pink are both Rapicauda species. The blue represents the island populations, and the pink are the mainland ones. And then that green represents the recently named species endemic to St. Martin. So just looking at this tree, we can see that it's pretty well supported along the backbone, and also that the island populations and mainland areas are reciprocally monophyletic. So we look at this tree and we can think about our hypotheses about divergence and dispersal, and we can see that it mostly supports the boomerang hypothesis. So you can see that the mainland populations are nested within the island ones. And just to give you a better visual of what this story kind of looks like. Here's the exact thing it came from. So first we have the Sonomensis on the southwestern distribution of the Amazon. And then they dispersed to the surrounding islands. And if you're wondering why the Panama Arc at the time of this divergence, that area wasn't connected to the mainland yet, so they were more, it's more accurate to call them like islands, because yeah, they weren't connected. Then next, we can see that they dispersed further along the Lesser Antillean Islands. And then there's that, there's the St. Martin named species. And then here we have, they came back to the mainland. And then we have a poorly supported branch of a population in Montserrat, and then further dispersion around the mainland to the Yucatan and Guiana Shield. And then nested inside the Guiana Shield populations are populations from Trinidad and Tobago. So I'd also like to look, this is the same tree, but now the axis on the bottom shows the time in millions of years ago when these divergences happened, and those error bars show where that divergence could have taken place. So we can see that the divergence between Sonomensis and the Rapicauda species was a really long time ago, at around 52 million years ago. And then the second oldest divergence is between Curaçaoan Rapicauda and the rest of its species at 39 million years ago. And just to think about how old these divergences are, we can look at the human evolutionary tree. So there's humans, and that second oldest divergence of 39 million years roughly correlates to the divergence of the human lineage to New World monkeys at around 38 million years ago. And it's kind of fun to think like Thecadactylus was, they're still considered the same species, but if you think about that for humans, that would be like considering humans the same species as something like howling monkeys. So next, I'd like to look at two more divergent states between Rapicauda and the most recently named species, and also the most recently diverged populations of Guyana and Trinidad and Tobago at 9 million years and 8 million years ago. And again, we can look at the human tree. So our most recent ancestor, we diverged from chimpanzees around 6 million years ago. So even though for Thecadactylus, those are its most recent divergences, that was still a very long time ago. Next, I want to look at the sequence polymorphism analysis for the ND2 gene. And this analysis basically is the genetic differences between populations. So first, we can look at the difference between Rapicauda and Sonomensis, and that's around 24% different. And then the second oldest divergence between Curaçaoan Rapicauda and all others is about 20% difference. Rapicauda and the St. Martin species is around 18% different. And then the most recent divergence for Thecadactylus between Guyana and Trinidad and Tobago is only about 8%. So again, if you don't really, you can't really tell what these numbers mean, the genome-wide difference between humans and chimpanzees is only around 9%. So to conclude, we can see that the tree supports our boomerang hypothesis and supports that island populations recolonize the mainland. So the Thecadactylus genus can be used as an example of islands serving as biological sources. And we can also see that despite their morphological similarities, Rapicauda and Sonomensis are very deeply diverged and have very high mitochondrial genetic differences. So future directions, this project is part of a larger study on the Thecadactylus genus. And so I worked only on the mitochondrial genome, but we're also working on using RADSeq data. So this is a preliminary tree made using IQ tree, and you can see that it has the same general pattern that the mitochondrial gene tree showed, so where the mainland populations are nested within those island ones. And so further genetic analysis can kind of further confirm the species description of that most recently described species on St. Martin. And then for further studying this genus, there's also a need to bridge this sampling gap because we don't have any samples from Colombia or Venezuela. So getting samples from there, seeing where they fall in the tree, it could kind of just help better understand the story of Thecadactylus's dispersal and also clarify the boundaries between those two mainland species. So finally, I'd like to acknowledge and thank all of these people and organizations, especially my mentors, Michael and Ed, the SSI directors, Lauren and Rebecca, Grace, Luigi, and all of the SSI interns. Thank you so much. And thank you for listening.
Awesome. Does anyone have any questions for Haley?
Why are they called turnip-tailed geckos?
So this picture shows it pretty well, but they don't have that tail originally, but when they fall off and regrow, it kind of regrows with this like little bump on the tail. So
I see the
why.
turnip. Thank you.
Yeah.
Thanks. What are their mechanisms of dispersal throughout the islands?
I'm not completely sure, but I'm pretty sure it's believed that herps disperse through like rafting. So like maybe just like bits of land break off and then they can disperse to like other islands like that. Yeah.
All right, anyone else? Anyone online?
Oh. Okay, this is connected to Curtis earlier. I'm curious, in the islands like research, is this something that is interesting to you all in studying dispersal? And is there a connection between studying the frogs and studying these cute little geckos? Like, is there something that's connected in like a further question that's answering science between islands for both of you?
Oh, yeah. Thank you for the question. So both of these projects were supported by the Islands 2030 Act that the Cal Academy has. And just to better understand the biodiversity across these islands, I think it's really important. Like, this genus wasn't really questioned until the early 2000s. And like, we never would have even known that there were multiple species until we did those genetic analysis. So yeah, I think just better understanding biodiversity and better understanding certain species is important.
So based just on the genetic data that you have, does it indicate that there's potentially more species than are currently recognized, or potentially fewer species than are currently recognized?
Well, if we look at the tree, we can see that since these divergences are super old, like I said, the various island populations were all reciprocally monophyletic, so pretty like grouped together. I think that since this is only based on a couple mitochondrial genes, there should be more genetic research and more research on possible morphological differences, because that really hasn't been like super looked into. So yeah, I'm not sure exactly, and I don't know if I'm really the presiding person for that.
All right, thank you so much, Haley.
Thank you.
All right. Next up, and our last presentation before a short break, for those of you who are needing one. Next up, we have Sofia Micci. from Cal Poly San Luis Obispo, and she was advised this summer by Laurel Allen.
Hello, everyone. My name is Sofia Micci, and I'm studying landscape architecture and sustainable environments at Cal Poly San Luis Obispo. And I have one of this summer's science communication interns working with Laurel Allen. As I began learning about what science communication is this summer, one of the first questions that emerged for me is how the physical environment we are in may influence our perception of science communication, specifically communication about conservation. From there, my research emerged, titled Act Naturally, How Physical Environments May Influence Receptiveness to Conservation Messaging. So coming into this internship, I had never worked in science communication before, barely even heard of it. And in school, I've spent a lot of time learning about the impact of the environment, both built, natural, and blended, on the people that work, live, and recreate in those spaces. According to the American Society of Landscape Architects, the mission of the profession is to design a sustainable and equitable world, and investing in global, social, and environmental change through the art and science of landscape architecture. As I switched gears into a science communication lens, I found these goals to still be very relevant. And as I began to brainstorm a research project, I knew I wanted to investigate what effect different kinds of environments could have on the effectiveness of science communication strategies. As I began searching the internet, I found lots of studies on the importance of human connection to nature and how being outside improves your health. I found how connectedness to nature encourages pro-conservation behaviors. And though my research does not survey connectedness to nature, these findings led me to believe that if spending time outdoors can improve your connectedness, and connectedness encourages conservation behaviors, then spending time outdoors may lead people to make more conservation efforts. This then led me to ask a series of questions to help drive my research. So my first question is an indoor or outdoor environment more conducive to influencing people to take conservation steps? So kind of in other words, does being in nature make someone more likely to want to protect it? Second, does the type of environment someone is in influence the type of conservation action that they're willing to take? So I hypothesized that if spending time in nature increases people's connectedness to nature, then receiving conservation messaging while physically in outdoor spaces or in nature, or potentially even in like a naturalistic indoor space, would also make someone more likely to take action to protect it. So kind of as I began my experiment, in order to test my hypothesis, I first had to find some distinct outdoor and indoor environments to test. So Cal Academy's Rainforest Dome offers me a really unique opportunity to test a blended environment, one that fully immerses you in living plants and animals, but that is technically an indoor space. In order to test conservation efforts, I needed a specific story to tell to the people I was testing. And in order to avoid the tired story of, like recycle, carpool, turn off the tap, etc., I wanted to settle on one specific species to study. So I settled on the southern sea otter, which I found to be a perfect candidate as it's native to California, it's widely known and loved, and it has a critical role in its habitat, being a keystone species. So sea otter conservation is, much greater than just the sea otters. It impacts the entire marine environment. So as I began, I started out with a pitch. So I created a script kind of explaining, starting with like sea otters are a keystone species in California's marine ecosystem, which means they play a critical role in the food web. So I shared that story with museum guests that I was surveying, and regardless of where they participated, whether it was indoor, outdoor, or blended, I shared this identical story, followed by an identical survey. So I asked 4 main survey questions to test my findings. The first one was, how likely are you to make household changes to support better water quality? How likely are you to make monetary donations to protect sea otters? How likely are you to volunteer your time for sea otter protection? And lastly, how likely are you to support legislation protections for sea otters? So these four questions in total kind of aim to cover most of the individual conservation efforts people can take without being too overwhelming or taking too much time in the survey. So our first space, for my outdoor space in the museum, I chose the living roof. So this space overlooks much of San Francisco from the roof of the Academy, and it's covered in thriving native California species, making it a perfect environment to test in. Here we have my blended environment. So I surveyed inside the Rainforest Dome at Cal Academy, and this dome is completely enclosed inside the museum, as you can see on the photo in the far left, kind of the outside of the dome, but it's incredibly immersive. There are skylights that provide natural lighting. As you walk through the space, you're surrounded by rainforest plants, insects, butterflies, birds, and the dome also sits at a constant 82 to 85 degrees to replicate the real rainforest, which was kind of tough to stand in at times. And then lastly, for my indoor space, I use the Naturalist classroom. So this is a space off of the side of the Naturalist Center, a space geared toward children with many museum specimens and learning activities about animals and our environment. The classroom itself is completely enclosed with no windows, fluorescent lights, and only some tables and lab equipment inside. So you can see on the far left is kind of your view as you enter the Naturalist Center. And then the middle photo is a look inside that classroom itself, where I did my testing. So over 5 hours of testing in each location, I surveyed 79 people on the roof, 99 in the rainforest, and 56 in the classroom. And I kind of, as I got my survey results in, was looking at the demographics of the people that I was studying. And so out of 234 total respondents, I found that women comprised 53% of all respondents, so slight majority. And I found that 75% of participants were under the age of 45, with the largest age group being the youngest group of 18 to 24-year-olds. So looking at a younger audience for sure. And I found also, interestingly, that the museum guests are a really highly educated group. Nearly 70% of the people I surveyed had an education above high school, so much higher than the national average. And I found that nearly 70% of California residents, a lot of people from San Francisco, from California, kind of almost the rest of them, other like 27% are not from California, but from the US, with then about 3% being international visitors. So now we're gonna start taking a look at my findings. So I'm going to walk us through 4 different graphs, one for each of my main questions, and they're going to show the responses in all 3 locations at once. So the blue line represents our responses on the roof, the green represents the rainforest, and the tan represents the classroom. So we can see a scale from 1 to 5 here, and 1 means they reported unlikely to take that conservation action, 3 meaning neutral, and 5 meaning they're very likely to take that conservation action. And as I looked through the data, I focused on which environments reported the most likelihood, so looking at the highest concentrations of 4s and 5s. And starting off with our first survey question here, which is how likely are you to make household changes to support better water quality, I was surprised at my results. The classroom showed the highest percentage of participants rating a 5, which goes exactly against my hypothesis, meaning that the highest likelihood of making the change. So, however, for 4, the other 2 spaces ranked higher. So things started off looking not quite sure what's going on here. Second, is how likely are you to make monetary donations to protect sea otters? And once again, surveying guest willingness showed highest in the classroom space. It was obvious on this one that monetary donations were one of the least appealing out of all the actions. You can see everyone kind of said 3. They didn't know what to pick. And out of all the environments, the classroom respondents still did show the highest number of 5s, but you can tell that this action is one that people don't really want to take. Here, it's how likely are you to volunteer your time for sea otter protections. We can also see here that the volunteer question yielded the most concentrated distribution of 1s, 2s, and 3s. So people don't want to do this either. The rainforest respondents identified as least likely to volunteer when we look in the 5 category. And this was the only action for which the roof earned the most 5s, which was interesting. The classroom comes in second with the rainforest third. One thing that I'm wondering from here is I'm wondering if, since volunteering, you kind of think of that as an outdoor action, wondering if because the roof is outdoor, maybe it's easier to picture yourself doing the outdoor action. So last question is, how likely are you to support legislation protections for sea otters? This one aligned with my hypothesis, with the roof in the rainforest polling the highest. We can also see that legislation is seen as the easiest or most appealing action, with the outdoor and blended environments showing basically identical levels of 4s and 5s, both outpacing the classroom. So some takeaways from these first 4 graphs are, our respondents seemed really a lot more specific to the type of action rather than a clear trend that was going to support my hypothesis. What was interesting is that the indoor classroom did show a higher or similar level of willingness, except for volunteering, which could be because it's an outdoor activity. And there were distinct similarities in preferences to different actions, regardless of which environment they're being tested in. So supporting legislation is seen as the easiest or most appealing action, with household changes coming in second, and volunteering time and monetary donations being seen as overall the least appealing actions. So, but wait, I decided yesterday actually to go back to my data and take another look at the responses in each environment overall, rather than separating them by each of the 4 actions. And interestingly, I had some very different results looking at the data this way. So I wanted to understand the overall weight for each environment for above and below average neutral willingness to make conservation efforts as a whole. To do this, I calculated the percentages for the total number of responses over all 4 questions together. Then I compared the responses that fell above and below 3 for each location. The 1 to 2 range represents the below average likelihood, and the 4 to 5 range representing above average likelihood. Suddenly, I can see that it's possible that people's strong feelings about individual actions were actually clouding my data, and maybe there really is something worth investigating further about respect to the physical environment. So now, though it's only by a couple percents, it does look like my hypothesis is being supported. You can see on the right that the roof had the highest percentage of above average willingness, followed by the rainforest, followed by the roof, which is what my hypothesis suggested. And then on the other side, you see the inverse, for below average likelihood, the classroom having the highest, followed by the rainforest, followed by the roof. So looking forward, as we've seen, my results were not entirely conclusive. In the future, I'd really like to explore some different ways to conduct this experiment to investigate my results further. First, it's seen that the preference to or aversion for individual actions really overshadowed my data related to the environment itself. In the future, I'd like to develop a new methodology that doesn't obscure crucial insights. One idea is maybe asking one more generalized question about conservation actions as a whole, rather than separating it into different categories. It would also be interesting to explore if different species have a higher or lower success rate with different actions, such as if the outdoor space is more successful to encouraging people to make outdoor-based actions. So second, the museum is already a really self-selecting group of people that really does not accurately represent the general population of the US or even San Francisco. My participants were highly educated, as I mentioned before, showing a 75% chance of an education above a high school diploma, where nationally that number is only 46%. So my participants also chose, they visited a paid science museum, meaning that they really are people that value science, knowledge, and education likely at a higher rate than the general population. So third, my indoor environment, the naturalist classroom, is really not a perfect example of an indoor space. The classroom is right off of the Naturalist Center, and while it's an indoor space, it has no plants or otherwise living things. It has specimens, photos, and interactive exercises on different animals, really blurring the lines between an outdoor and a blended space. As I began my surveying in the classroom, I also learned that there's a surge of visitors every single hour coming straight from the planetarium, which shows 3 different shows, one of which is titled Habitat Earth and tells a really similar conservation story to the one that I was telling. So they just came out of a really immersive exhibit on a similar topic, potentially really obscuring my results. And obviously, though the planetarium is indoors, it's, people just came from it and it's really immersive. So the rainforest also is not a typical blended space. It's a really unique experience at Cal Academy, and it's climate controlled, it's really hot and humid, which also could have impacted people's willingness to do things like physically volunteer when they're uncomfortable in the space they're in. A space like an atrium, something with like native plants, maybe a more like neutral climate, could potentially do a better job at looking at a blended space. Looking forward, it would be really interesting to conduct kind of the same experience, but with participants from all over the city of San Francisco, not just museum guests. I'd like to find an indoor space further separated from other conservation messaging, and one that participants could potentially spend more time in as well, to see if longer time you spend in a space improves your action or otherwise. I predict that doing these things would let more trends kind of develop more obviously with a more diverse group of people. I also need more data in general. Though I surveyed 234 people in total, having at least 100 participants in every space would really give me a much more statistically significant look at the overall idea. So yeah, looking forward, a lot of changes I could make to see some more concrete results, but things are looking really interesting at supporting my hypothesis and where we could go from here. So, thank you. Specifically, I'd like to thank Laurel Allen, my mentor, as well as program directors, Dr. Lauren Esposito and Dr. Rebecca Johnson and Dr. Ed Myers, as well as the entire digital engagement team and everyone that helped me with all my surveying and my project. Thank you so much.
All right. Thanks, Sophia. Does anyone have any questions?
I was just curious, the times that you collected your data, was it the same in each location? And what times did you do it?
That is a good question. Not exactly the same. That was something I looked at. Obviously, eliminating all the differences is a good thing. In the rainforest, I surveyed between like 10 and 1 p.m. In both my slots. In the classroom, it was between 11 and 1:30 or 2, so pretty similar. And then on the roof, I did it in the afternoon, so between like 1 and 4 for both my spots.
Yes, it will be interesting to take this experiment outside into the city, but not only test for different diversity, but different community sites, because they have their own perspectives. So it'll be interesting to find out what the results would be.
Yeah.
I was wondering if you looked at the responses from like a demographic lens, if there was like a difference in responses based on age group and gender, and if that played into people's willingness.
I unfortunately did not have time to break it down at that level for this, but that is definitely something I want to look at in the future. Kind of like a sub hypothesis I had is I'm wondering if things like differences in space might affect younger people more. But yeah, it would be definitely interesting to look at that in the future.
Hi, so I just wanted to know if you added an aspect of if they ever learned about the outdoors and their environments while they were growing up would affect the data in any way.
Yeah, that would be interesting. And I think, looking at that as well as like how much time people spend outdoors on a regular basis, I think would be interesting to study.
I had a question about like going to sort of like the phrasing, like you mentioned, like your willingness to like donate. I wonder if you added like asking more about like how capable you feel of like donating or like committing your time to volunteering. Cause like, I wonder with the volunteering question, if younger people maybe don't feel like they have enough time, but then maybe older people feel like they don't have necessarily like the ability, even if they're like have time because they're retired. So I think it'd be interesting to see like if like capability, like how people feel like empowered to make change. I guess that's not a question, but.
Thank you.
Yeah, I had one question, Sophia. I wonder if, in that classroom space where you're like kind of pulling people aside, it feels like a little special in maybe a way that the others don't, right? It feels like a little more exclusive than like walking by someone in the rainforest. And I wonder if that plays into someone feeling like extra special to be in the survey.
Interesting. in like more likely to make a positive.
More likely
Yeah.
to have a positive because they're like, I was chosen to be in
Maybe.
this special
What was
place.
interesting is that actually I think it being separated made people more likely to want to participate. Whereas in the other space, they were more likely to come to me rather than seeking them out. So I did have people in the classroom where I would ask if they wanted to participate and they would say no. And that happened less likely in the other spaces. So yeah, I'm not sure how that would affect things.
Yeah, super interesting. Oh, there's a question online. Hold on one sec.
Hi, Sophia. Jacques Johnson asks, why do you think science communication is a vital component in the field of science?
That is a good question. Yeah, I mean, it's really important for, not just me, but for all the interns. Like I mean, that's what everyone's doing today is science communication. But like, if you do your research, it's really important to share with other people to build the research and also just to, get everyone else passionate about it. So I think it's a really critical aspect to bring science out of the direct science communication or direct science community and bring it to everybody. Really important.
All right. Well, thank you, Sophia. All right, everyone. So we're going to take a break. It's going to be a little longer than scheduled so we can be on schedule so people know when to come back. So 2:40, 20 minutes, back here at 2:40. We'll see you back online then as well.
How about now? No wonder nobody could hear me before. I'm just yelling into the void. All right, next up we have Chantal Watson, who's joining us as an alum of Rochester Institute of Technology. And she was working this summer with Dr. Shannon Tushingham. Come on up.
Thank you, Lauren. Hi, my name's Chantel. Yes, I graduated from Rochester Institute of Technology. Last year, I studied medical illustration, and now I am the biological illustrator for our interns cohort. And I am doing a study with Dr. Shannon Tushingham in anthropology on a deep dive of baskets and just investigating how it is a cultural artifact as well as an ecological artifact through my illustrations. So for the basis of my entire presentation here, I'm going to be going over the illustrations that I conducted over this whole summer, going into how I visually analyze these baskets, especially in California, and understanding how these different tribes of California diversify themselves from these kinds with their own cultural identity based on baskets. And then I will also go over some flyers that I created for outreach for anthropology, the anthropology department here at the CAS, and as well as Snapchat Kyle Coast. So first, before I start on the basketry, I'm going to discuss visual media and how it's important and really significant to what I'm doing here. Visual media is something that's extremely important with illustrations. And I think highlighting what that means and why it's significant in places with cultural and historical subjects is something that should be investigated. But for the main reason, these are making complex concepts accessible to the public. People are visual learners, and I think you have to always have your audience in mind when you are creating any kind of work. With visual media, you want your research to be easy to comprehend, and for those who have no prior background knowledge on whatever topic you're discussing, you want them to be able to understand that. And so visual media is something that can be a great use, whether it's infographics, photography, illustrations, even videos. Those are all types of media that can really highlight these kinds of concepts. And especially with what I'm working with anthropology, this can be a means of preservation and just how this is conducted through this kind of media, especially with objects and artifacts. A lot of stuff in the collections are very precious and very gentle. So you have to be very careful with it, but being able to memorialize these objects through art or even photography in turn preserves them and their practice and the different techniques that made them significant to these cultures. You want to have visitors understand, not only look at your work, but understand what they're looking at. So it's a means of education. So being able to see this visual media and understanding that there's something to learn from it, which is why I conducted my work with infographics, is a big part of why you want to have this media done. And it works in tandem with promotion of conservation and land stewardship, which is what I'm focusing on completely, and how I can use this as a means to educate a wider audience, especially here at the CAS, how it has an audience that can be younger, but also those who are more interested in science. You don't see a lot of science museums have highlights of indigenous practices here. So I think when done correctly, this is something that can really highlight and advocate for these kinds of subjects. And so that goes into the State of Nature exhibit, which I think highlights that completely well, and a big inspiration for the illustrations that I've done this summer. I think with the illustrations that the State of Nature has, they highlight different areas of indigenous practices and land management. Through illustrations, they've hired indigenous people to work on this, and just collaborating with those people and having something that is not only beautiful visually because it is amazing illustrations, but being able to communicate that in a way where it's able for people to walk through these exhibits and be interested in what they're learning rather than a very like primitive and archaic way of showing these, because these are subjects with people who have cultural backgrounds, traditional backgrounds, and you want to highlight that in a way that celebrates it rather than make it seem archaic. And so with that, my objectives for my project for the summer was to be able to acknowledge these kind of practices through my artwork, and just be able to highlight basket weaving in Indigenous tribes from a perspective that can be from a modern take, because lots of museums, especially natural history museums, do not highlight this in a way that you see at Cal Academy. So being able to do that with my work, I want to be able to show a modern take that can be presented in museums at exhibits, even though that would be impossible to do now because it takes a while to make exhibits, but a good idea of how it would be. And also I want to tie in traditional ecological knowledge into my work and create a correlation between how basketry can actually be a means of like land stewardship, and also just highlighting the beauty of like the different techniques that different tribes conduct with these baskets, and highlight and celebrating it because they are amazing. So my methods for this was very bare bones because it's just me doing artwork. But I was able to go to collections for various sections, not just anthropology, which was amazing. So I was able to like go into collections for botany, and hopefully if I were to continue this, I would have gone to like ornithology because there are also some ties there with anthropology. But just being able to go there was really helpful to conducting what I wanted to do this summer. And just having conversations with local Native communities was something that was a big part of my work, and just talking with people and getting firsthand experiences is something that is very important when creating any kind of work regarding a group of people that I'm not a part of. And just the various art mediums that I use for my work. I work traditionally and digitally, so I usually sketch my work by pen or pencil and then move it digitally onto my iPad, if that counts as a method. So I'll go into why I explored the collections, mainly just anthropology collections and a herbarium. A main reason as to why I wanted to go into these collections was to get firsthand references for my work. I love to work from life. I think the best way to illustrate any kind of subjects are working from life. And just seeing the objects in front of you, you get a real good idea of the form of what you have here. And there's just details that you cannot see when you go on a computer that you really just can be highlighted when it's right in front of you. So these are images of me taking baskets out of the collections for anthropology, which was very great to handle, very nerve-wracking to try and see these actual baskets like right in front of me and being able to take it out. And just how gentle they are and how like there's so much care and love into each one of them. And you can tell just how important they are to the different tribes that created them. Because especially even this one, a lot of them, as you'll see, gift baskets have very like intricate pieces added to it, feathers or glass beads. So you really want to be careful when you're handling those. And I also worked with Emily, who is the curator for botany collections, and she let me have access to the herbarium, where she gave me some plants that have correlation to basket weaving. These are some plants that were used for these practices, and primarily, and I will go into depth about the significance of most plants in basketry. But these are some plants that like I got to see firsthand that I wanted to understand, have a good feeling of how these were created into the baskets that I had in the last slide. And just understanding that how these plants were like cultivated to be able to create vast amount of like baskets and just different sizes, and the amount of like work that goes into it is just amazing to comprehend. And so these are more plants here, and these are my personal pictures. The last one was Emily. What Emily had was a display of basket weaving plants in the herbarium and showing off which ones were used. These are just a range of images that I wanted for myself to use for my illustrations as a reference. Yeah. And so we'll go on to like the sketches and thumbnails, because I think that's very important. You can't just get into creating an illustration. You have to actually lay out what you want to do, and like the idea to really have a good concept of what you want illustrated and just the different ebbs and flows of how it can come together. So these were my initial layouts and ideas of how I wanted my work to look. I went through a multitude of like different ideas of just how I wanted to lay out everything. But in the end, I knew that I wanted to create infographics. So this is just a general layout of highlighting the plants that are demonstrated on there, and just highlighting the different regions of where in California that I want to show, as well as which types of baskets. I initially started with the idea of having an infographic and flashcards. And then the flashcards idea got booted because I didn't really want to do flashcards, and I thought that infographics would be more appropriate for the take that I wanted to have on how this can be shown in modern museums besides here. So this was more of the exclusive layout of what I was to do, and I like to sketch out the initial basket of how it would look. This is me working traditionally and digitally, and just having a good idea of what I want the layout to be, what information I have. Even though there's not real words there, I know in my head what I want the words to be when I make the actual illustration. So this is something that will get me going to get to the actual final illustration, which is here. So this is an infographic that I created on burden baskets, which are baskets that were typically used by many California tribes here. And they use these baskets to be able to hold storage for and gather and harvest plants and different kinds, just acorns, plants, any kind of harvest. They store it in these baskets. And the materials were used here is highlighted with the different kind of roots. There's white root, there's willow shoots that they use, and these are all just highlighting of the herbarium references that I got and using that within the illustration. And then underneath here, I wanted to still highlight how these plants were cultivated and the management that goes behind how these are made, rather than just having random baskets on the page. But I have two different tribes. This is a Klamath River, and then this is a Wintu burden baskets, and which are very different in shape. And I think it's very obvious to tribes as well, because they want to differentiate themselves by the styles that they have, the techniques that they use, even the material that they use. This basket is like a cone shape because it's usually meant to hold firewood and oak, but this one is not cone shaped. It's more just a regular looking basket. So there's a lot of like characteristics that these will have that really sets aside how these tribes can differentiate themselves with one another. And so another one that I did was the gift baskets. And this is just a brief infographic of just what they are. And I thought this basket was beautiful. This is a Pomo gift basket that used California quail top knot feathers. And so that's the feathers on top of their head. And this uses glass beads as well. And most of these use the same exact plants that are used. It's usually sedges, and different type. I have a slide for it, so I will get to that later. But different materials that are used are usually found the same in all of the different kinds of baskets. And I really wanted to highlight just the area of where these gift baskets are typically from, because I think these are such significant and very small baskets. And they can range in like sizes from like larger to even like as small as like the palm of your hand. Some are fully covered in feathers. And it says here that fully feathered gift baskets were exclusively made in Northern California tribes. So this would usually be the region that you'll find it. Pomo, Coast, Miwok, Wapo, and Patwin tribes are usually the ones who would have fully feathered gift baskets. And gift baskets were usually given as like peace offerings to one another to strengthen ties and relationships with one another, as well as they were gifted for familiar bonds. And also sometimes they were buried with those who owned the basket. So they do have very like cultural ties, like it's very strong and it very like respectful artifact that it's used. But these are the illustrations that I would want to show in an exhibit that was highlighting baskets. And then this is a seed beater basket, which would have been with used with a burden basket for collecting seeds when you're out in a harvest. But this one would have been on the burden basket slide. So this is just on its own as a solo illustration. So here I have, my notes are not there, but I'll just do it off my head. But these are the main proponents of materials that are used. Most of these beargrass, deergrass, tules, and sedges are the materials that are used in baskets that are usually foundational in most of them. These are always like pruned and propagated by native people. They are done with lots of fire management, especially sedges. Sedges grow by water. And so because they're near water, it takes a lot of like management of the roots and the rhizomes, which a lot of plant parts are incorporated in baskets and within the plants that are used. So it's not just like the roots that are used, but it's the rhizomes, the stems, the flower stalks, all of those are like propagated in a way that can be used within the baskets. And the management of this also helps these plants for years to come, especially with like traditional burning. The act of burning some of these plants actually helps like the stimulation of like new sprout growth and seed production. So many of these actually and these are done by basket weavers. So they take these methods of how to manage the land. And in turn, they help the ecosystem that these plants are cultivating in. So I will move on to the outreach flyers that I did for the summer and just highlighting what those were for and what they came out to be. So first, I did a flyer for Snapshot Cow Coast. I went to a Native Arts Festival this summer, and that was something that we wanted to outreach to people there and just the local communities and let them know what we are doing about biodiversity and just wanting to collaborate with these communities and being able to use the iNaturalist app and identify species that are out there. So this was a big thing that we handed out when we went to this festival, just to have people come out and be able to interact with us and talk with us while also promoting the habitat. So this is the flyer that I created, and I actually did use the iNaturalist app for references for the abalone, and these were the high in demand abalone that were for Snapshot Cow Coast this year that wanted to be identified. But I worked closely with my mentor Shannon and as well as Julianna, who did the layout of the text as well as writing the text, because I'm not going to write the whole thing. I'll draw it instead of write it. So the majority of this had a lot of like refining, and many like drafts were made for this. But this isn't even the final draft yet because there's still some revisions, but this was like a great way to like interact with people out there and tell them that they can come and chat with us while we're tide pooling. And we also went to the Gorman Museum this summer And this was at UC Davis. And a big thing for me when I went was that this is a contemporary art museum for Native communities. And so it really highlights just the work and a lot of like creative freedom that we're giving to Native people here represented. And I think something that stuck out to me a lot was the Northwest Coast art style, which is very distinct and has very lots of negative and positive space and a limited color palette. And this was just the first thing that I saw when I went to that museum, and I almost made my full project just investigating this art style. But I think it was so beautiful, and it's just, there's so much like significance with like the animals that are incorporated with it. Many of these paintings tell different stories based off of how they're done. And there's just a strict guideline to how like these illustrations are created. On face front, it looks like it's just a very simple like thing to create, but there's so many principles and like characteristics to this work. And so I did a study on this style of Northwest artwork and just the use of like different shapes, and it's very specific. And then I found a guideline book on how to create it. So you have to start with like an illustration of an actual animal, and then from there you have to simplify it completely down. But the use of like different shapes like ovoids and just the distance between a lot of objects that you have to keep in mind of everything. And so it looks like it can be super simple to create, but there's just a lot of principles that they follow, and they follow it completely easily. But for me, this is something that I was very focused on just how the lines were parallel, and just like what color schemes could be used. And so after doing this study, I really wanted to be able to incorporate that with my work. And so this is our outreach poster for Indigenous tribes to come to the collections. And this was also given out at the festival just to be able to show them what we have and encourage them to come see the collections that we have in anthropology. So I created this salmon, but also, which is very hard to do. I think this took me a few days, and just being able to use these practices, I think it was something that really like took me out of my comfort zone. But I really as I like started to do it, I understood like how this was done. And I think it's just a beautiful art form. But yeah, in conclusion, I would like to do more illustrations on this, on more baskets and even like food sovereignty. But I think this was a great experience for me. I'm not really knowledgeable in anthropology as I'm a medical illustrator. But this was such a unique experience that I've never got to see. And I'm very grateful just for working with everybody here. And so my acknowledgements here are Dr. Shannon Tushingham and Laura and just everyone in the anthropology department. And then thank you to our program directors, Lauren and Ed and Rebecca, and then all my friends here that I made. Yeah, so thank you.
I think we have time for maybe one question as we switch out. Thank you so much, Chantel. Awesome.
Has learning more about these traditional Indigenous art forms influenced that you push your own art elsewhere in life?
You know, I was thinking about it as I was here this summer, and I think working with museums, especially just with this Indigenous work and how I felt like I was in turn advocating for these kind of groups. And I think that's a main part of my work. I love to advocate for groups of people who are not typically represented in work. So I feel like, especially in a museum, a natural museum would be something that I would pursue creating illustrations and even doing exhibit design for in some point. So we'll see. We'll see.
All right. Thanks, Chantel. All right. Next up, we have Emilio Chavez, who joins us from the University of California, Berkeley, and he worked with Eric Steiner and Lauren Esposito this summer. Come on up, Emilio.
All right, hello everybody. Osio, danke doa ustiona. Greetings everybody. My name is Emilio Chavez, and over the summer I've had the privilege to work at the California Academy of Sciences Arachnology Lab with my mentors Eric Steiner and Dr. Lauren Esposito on developing a new non-lethal method to analyze scorpion diets. So before we begin, let us talk about why scorpions are so important. Well, essentially, they are a relatively understudied group, and we don't know too much about their diets. So recent research shows that we know that they are generalist predators. They prey upon vertebrates and invertebrates, and even other scorpions within their own species, so they are cannibalistic. In addition to this, we know that they have the highest biomass out of any other animal within desert ecosystems, meaning that there's a lot of them out there that are eating a lot of things in addition to being consumed a lot as well. They are a good food source for other vertebrate species. And since they are both apex invertebrate predators and both prey, we believe that they play some sort of key factor in the trophic systems within the environments in which they inhabit, which makes them so important for our studies here today. So our studies have been conducted in Baja California Sur in Mexico, which is a relatively diverse area for scorpions. Within the region we collected, there were 16 different species. However, in our specific region here, it's located in San Juanico. There were 11 known species at the time, most of which that were collected were either Centroides or Parajovius. I'm still getting the name in my head, apologies. But the last study that had been conducted in this region was actually in the 1980s to 90s by Gary Polis doing some cornerstone ecological studies. So we have on this map here, you see at the top outlined in the green, you could see this completely different region down in the bottom at the Magdalena Plain. And you see San Juanico right in the middle with this white outlined region. So essentially our study site is at this sort of funnel of biodiversity, if you will. Basically a meeting point between these two different regions, which promotes high biodiversity between the two scorpions that inhabit each region, which makes it a relatively important area to study. And this brings us to our overall goal, which is to develop some type of new non-lethal method in order to sample the regurgitate from these scorpions and to assess their diets, thus giving us a better understanding of their trophic interactions and potentially their importance within their environments. And once taken over a longer period of time, perhaps their diets can give us a better understanding of the overall health of the ecosystems of which they inhabit. So the stages of our project were broken up into two separate ones. One was conducted last year, actually, down in Baja California Sur at San Juanico. And this was basically the development of a standardized method to extract the regurgitate from the scorpions. And this was taken through the idea that was actually created for some beetle species. And it relies on the idea of heating the specimen in order to basically force regurgitation. While stage 2 occurs here at the California Academy of Sciences with my work, and that essentially is more lab-oriented, and it focuses on the successful amplification of the DNA within each regurgitated sample. That way we could submit it for amplicon metabarcode sequencing, and we could possibly see all of the potential species within the regurgitated samples, thus giving an idea of some potential diets. And our method was adapted from some previous studies that have been conducted with spiders specifically. So it's kind of still with arachnids, we're trying to make it work here with scorpions. So this brings us to the concept of DNA barcoding, which is essentially a way to ID species by utilizing these short segments of DNA, which we call barcode fragments, essentially. And these segments of DNA are unique depending on species. And you can see the process of this here in this figure. You can see we have some sort of unknown organism, and we have to extract our DNA, and we have to amplify it using some designated primers that are specifically made to amplify these barcode regions of DNA. Once we get this DNA sequenced, it comes back to us in the form of typically some nucleotide bases. And we could then use a reference database, such as NCBI's GenBank. We could use that and blast the resulting sequence to potentially find a percentage match, thus giving us an ID on the DNA we extracted for species. However, most studies that are conducted for modern diets in insects utilizes the idea of DNA metabarcoding, which is essentially uses the same concept of barcode sequences. However, instead of doing individual samples at a time, we can combine all of our samples into one larger homogenized mixture. We could then amplify every single bit of DNA within the mixture and sequence it all at once, thus resulting in a massive line of sequences, which we could use a reference database for, and potentially identify every single different species within our sample. So it does more simultaneously, and it takes up less time than DNA barcoding. And previous work has been done on this using bat feces for other arthropods. And as I mentioned before, it also has been used for some spider studies as well, their gut contents. The only main issue for this is that for arachnids, it's a relatively lethal process. You have to either remove the stomachs or lyse the whole organism, which you kind of need a stomach to live. So it's a pretty lethal method. So we'd like to keep that in there, preferably. So this brings us to our concept of amplicon sequencing. Amplicon is a type of metabarcoding. As I mentioned before, it essentially takes a larger homogenized mixture and sequences it for pretty much all of these individual genes. So if we look at this in stages, I know there's a spider on there. Imagine that's a scorpion, okay? So when it says individual DNA extraction, consider that stage 1 of this project. That was conducted down in Baja, California, where they developed a way to extract, regurgitate, and extract DNA from it. And then you see that BARC, PCR, and indexing and the sequencing. That's essentially stage 2 of this project, which we are partially the way through right now. We have successfully found a way to amplify our DNA and undergo PCR. And we also have a reference genome for all of our samples for the predators themselves, the predatory scorpions, which we could then use for the sequencing here. We could filter out the predatory DNA from potential prey, thus making it a little easier to identify what we're looking for. And this is actually how the regurgitate is extracted from our samples. So you see on the far right over there, you see that heating chamber. Essentially, these scorpions are placed live inside of a falcon tube, which is a relatively thick plastic tube that's then immersed in boiled water. And it rapidly heats the air around the scorpion to around 80 degrees Celsius, thus forcing the scorpions to regurgitate. You actually see their chelicerae kind of heave forward in videos of it. And at this point, it's like a regurgitory reflex. Once this occurs, either the scorpion will pretty much vomit or it will produce nothing. And if that's the case, either way, the scorpion won't do that reflex again and pretty much it's done. And if you're lucky, you will get this extracted regurgitate here. You can kind of see it in the bottom. It almost looks like water. It's a very clear substance. However, the color and texture does vary, which you'll see in the next few slides here. But then you place these scorpions in these resting chambers, which allow for airflow, and it also contains some food. That way they could replenish their nourishment since they just pretty much purged it all. And once that is completed and they cool down and they regain their strength, they're released in the same location from which they're extracted. So it essentially prevents their death, but it also, doesn't displace them. So it's a relatively good method to use. These are 2 examples of different samples here. You see the one on the right is just as the previous one was. It's relatively clear. The one on the left is pretty murky, has a lot of particulate, and it's kind of brown. Now, the reasoning behind this is potentially because scorpions have a really low metabolic rate. They could potentially go for months at a time between meals. So it is possible that sample 60 over there maybe ate a couple months prior to being sampled. So it could be at a different stage in digestion. Meanwhile, the one on 61 is pretty murky and has a lot of particulate. So maybe it ate a week ago and it's at a relatively early stage in digestion. But in total, 36 samples were extracted down in Baja, California and submitted up here to the California Academy of Sciences for me to work on for stage 2. And this brings us to stage 2 here. There we go. So pretty much my entire goal is to figure out a successful way to amplify our DNA that way we could use metabarcode sequencing. That has been the entire goal so far for stage 2. And as you could see, the steps here, DNA extraction, that was completed during stage 1 in Baja, California. So that part is already completed. And my part in this so far has been to successfully amplify our DNA, which has been done successfully now. So that part is completed. And that only leaves one more step in order to properly identify potential prey and diet. And that's for amplicon sequencing, which we are hoping to complete in the very near future. As you can see here, just outlines our area here. And this brings us to some potential problems that could occur during our process. Since this has never really been done before for scorpions, we never knew what to expect. We had no idea what could go wrong, but we had some basic ideas. We had no idea whether our regurgitate method would work. So there was always a chance that there would be no DNA present within our regurgitant, or perhaps the DNA was too degraded due to the stomach enzymes of the scorpion, perhaps. Or maybe there was some type of inhibitor present within the stomachs of the scorpions that would prevent it from being amplified and making it unsuccessful. However, we were able to use some scientific instruments called a nanodrop and a qubit, which measure the quality and quantity of the DNA present within each sample. And based on those values, we were able to determine that there was in fact DNA present within our samples. And it was in fact a quality that was good enough to amplify. Therefore, the first two issues should not occur, should be perfectly doable. However, according to our nanodrop values, we were able to determine that there was the presence of some sort of protein-based contaminant within our samples, which had the potential to possibly inhibit amplification. And our strategy overall for stage 2 was to just do a huge amount of trials, do as many trials as possible with as many different primers as possible that were targeted for arthropods. So essentially, we used 4 different primers for our samples. We used one called mini barcode, which amplifies the CO1 mitochondrial gene. Another one called hexapod 12S, which amplifies the 12S ribosomal gene. One called Chir16S, which amplifies the 16S ribosomal gene, and another one called hexapod CO1, which once again amplifies the CO1 mitochondrial gene region. Each one targets a different area, so maybe if one area was damaged for one sample, it had the potential to have the area successful for another primer, thus allowing maybe certain samples to amplify under different conditions. They're relatively inconsistent since it's vomit. None of it's the same, really. So each sample was relatively unique. And out of all the attempts we made on our initial trials, we only had one successful sample that was produced using our hexapod CO1 primer here. As you can see, it's not exactly what you want. The band there, where the red arrow is relatively smudged. It suggests that amplification was partially successful, but it's not the quality of which you would like. If anything, we would like to have a very crisp, clear, defined band with no smudging whatsoever. However, this does inform us that the primer itself is bonding correctly to DNA, but something was inhibiting the amplification itself. And this was also occurring in our other primers as well. In our other areas here, you can see similar smudges, however, no banding present. So DNA is there, but it is not being amplified, essentially. So this allowed us to, this basically gave us the incentive to create a brand new method that's centered around different ways to enhance amplification, essentially. It was around the idea of DNA dilutions, which could potentially inhibit the amount of protein inhibitor in each sample, thus increasing the probability of the DNA amplifying successfully. We also increased the amount of VBSA in each sample, which essentially increases the yield of each potential amplification cycle. We then decrease the amount of annealing cycles in each round, and that essentially decreases the amount of overall DNA amplified. And this acted as a way to reduce the bias within each sample. Since this regurgitate did come from a scorpion, it was most likely that the DNA within each sample was just pure scorpion. So decreasing the amount of annealing cycles reduced the amount of scorpion DNA, and thus promoted the probability of potentially detecting prey. In addition to this, we also modified annealing temperatures during PCR in our thermocycler. We made this decision essentially because we ran some temperature gradient testing on our primers, and we found that there was an ideal temperature region for pretty much all of our samples to run, and that was around 47 degrees Celsius. So we decided that we would make that our annealing temperature. In addition to this, we also used a higher fidelity tack, which would essentially just produce better quality samples during amplification. And overall, it was a great success. We had successful amplification for both our mini barcode and our Chir16S primers. So we were able to successfully amplify the CO1 mitochondrial gene and the 16S ribosomal gene in almost all of our samples here. You can see that very clear banding, and that's exactly what you want to see. That was a good day. Everyone was kind of shocked with that one. And that brings us to our overall development. Out of all of our samples, we were able to successfully amplify 35 out of 36 for our 16S region, in addition to successfully amplifying 8 samples for the CO1 mitochondrial region. And since we had a majority of the 16S region, we were able to blast our resulting sequence that we got. We submitted it for sequencing, and we then used the resulting sequence to input it into a database. We found that the most closely related species to our sequences were scorpions. And we then had photo IDs of each scorpion that had the original sample taken of, and it matched the species of the scorpion images. So therefore, we were able to make a conclusion that we most likely had the DNA of the predator that we could then use as a reference to filter out for potential prey. And this would be done during amplicon sequencing. And then using our 2 areas here, we were able to use our sequences to create sort of this tree here, just to see that there are in fact 2 different species. You can see Centroides here as well as Vohodius, apologies. You can see just that clear delineation there, just showing that there are 2 different scorpion species. And this brings us to our overall main lessons of this study, which is essentially we were able to develop a standardized, non-lethal way to extract regurgitation. Stage 1 has been completed successfully. We are also able to successfully extract DNA from the regurgitate itself. And that was a relatively big success. And the DNA itself is usable, which is a relatively big deal. In addition to this, we were also able to obtain the 16S genomes for all of our predatory scorpions as well, which is relatively big since it allows us to make amplicon sequencing a little easier from filtering out potential predatory DNA. And this brings us to our next steps here, the future. What we're trying to do is we're trying to do this meta barcoding, essentially. And in order to do this, we're going to have to undergo a special method called 2-steps PCR. And essentially what this does is we're amplifying these barcode regions of DNA. And then we have to undergo a secondary PCR, which attaches these tails to the ends of this DNA. And these adaptive tails allow metabarcoding to take place, essentially. So that would allow us to combine all of our vomit samples, all of our DNA, into one large mixture, which we could then sequence all at once. And then this is where that predatory DNA comes into play. We could then filter out all the resulting sequences for predator DNA, thus leaving only prey to be detected. And we are hoping we could get this done within the next few weeks. And if so, we could possibly have a new way to determine scorpion diets through a molecular analysis. And it's very important that we actually do this. Scorpions, they play a very key role in their ecosystems. We just don't fully understand their trophic interactions. As both predator and prey, they play a key role in basically maintaining the balance within an environment. And that should definitely be studied in the future, and it should be maintained. And that brings us to the acknowledgements here. I'd like to thank everyone, Dr. Lauren Esposito, my mentor, Eric, in addition to the whole arachnology lab, the CCG team. Pretty much none of this would have been possible unless everyone was here. We didn't know what would work. We were just bouncing ideas off of each other, things that worked for us in one sample, something that worked for someone else in plants. Pretty much, we were trying anything that could potentially work. And none of it would be possible without everyone's amazing ideas. So thank you all, and are there any questions?
Thanks, Amelia. I think we have time for a couple of questions.
The hope is to use this method for other critters to extract the DNA from their bellies and then find out what their consumption is?
Currently, it seems like I'm not entirely sure on this. It's possible that the stomach contents for each species could be different. If this works with scorpions, it could become a standardized thing that you use a baseline method for scorpions specifically, but I wouldn't say it could be used for every other species or every other arachnid. I'd say potentially it's going to have to be tweaked a little bit for each different species, but it could become a decent just baseline idea of what to expect.
You had mentioned earlier that there was something interfering with the PCR, with a protein that was interfering with that. Do you think that possibly that protein could be from the
diet of the scorpion, so something that they ate, and that possibly could be a protein from their diet that is interfering with it? And if so, do you think that controlling what they ate would possibly fix this problem more?
Potentially having an idea, if we keep them in captivity and we feed them a predetermined diet, we would know what to expect from the regurgitate. So if anything, we could use that as a good control. Whether or not this protein is from whatever enzyme is in their stomach being produced, or potentially from their prey, we don't exactly know at the moment. We don't know what protein it is, or if we know where it is, maybe we can know where it comes from. But since we do not have that knowledge, I can't exactly say if it's from the scorpion's own stomach or potentially some type of pre-digested prey. So I'd say a little more research is needed to make that determination, but it's something good to look into.
All right, last question from Carol.
I know you mentioned like filtering out the scorpion sequences, so you'll only have like the prey. How would that affect your results if you mentioned that the scorpions are cannibalistic and they do eat other scorpions?
So since scorpions are cannibalistic, that makes things a little more tricky. Essentially, would have to go far beyond the species barcode sequences, and we'll have to move on to something a little more defined to individuals within a species. And those use these very specific snippets of DNA. I'm not too entirely sure in the process, but I know we'll need the original predator, and we will need to have something very specific from the prey in order to determine that level of identification. And I'm not too entirely sure where that takes us, but maybe if I read some more, I could figure it out.
Awesome. Thanks, Emilio.
Yeah, thank you.
All right. Next up, we have Audrey Garcia, who is from UCLA, and this summer worked with Dr. Nat Low and Dr. Avery Hill.
Right. Hi, thank you, Rebecca. I am Audrey Garcia. I'm the community data science intern here at SSI. And like she said, I worked with Dr. Nat Low and Dr. Avery Hill. And my project is on investigating shifts in geographical species distribution as a result of reservoir construction in the northern Sierra Nevada foothills. So I first wanted to start off by talking a little bit about what data science is. So like other sciences, it is an iterative process. So it doesn't necessarily follow one linear process of how you do it. But first, you want to begin with data collection. And this typically comes from existing repositories or data sets. Then you want to process that data that you get and refine it so it is usable to you and like follows the filters that you want it to. And then you go on to analyzing that data, asking questions about it and trying to gain some insight from it. And then finally, you'll want to communicate this data and communicate what you found. The other field relevant to my project is community science, which seeks to connect people to science, nature, and each other. And the way we do this here at the Academy is through BioBlitzes, challenges, and events like Snapshot CalCoast, seen here. And the purpose of community science is to crowdsource biodiversity data across levels of expertise by including non-scientists and scientists alike. And the way we do this is through the app iNaturalist, which is just a free app that you can get on your phone. Anyone can, and you go out and you take pictures of any species that you observe, and it will document the time, date, and location of it. And if you get 2 species identifications and you meet those previous criteria, then your observation can be considered research grade. So once it's research grade, then it can be uploaded to the Global Biodiversity Information Facility, or GBIF. And GBIF is a massive online data set of biodiversity data that is free and accessible to anyone. So we can see here that there's a bunch of human observations that come mostly from iNaturalist, but we also see there's a bunch of preserved specimens that come from museums like here at the Academy. So with all of this massive amount of data, I knew I wanted to look at California and questions about biodiversity in California, but this is very general and vague, so I just wanted to follow what my interests were in this. So my interests were about California history and the land use changes that we see throughout it. And if we're talking about that, one of the most drastic changes to California land was through settler colonialism and the resource extraction that was driven by that. And that is mainly the gold rush and mining that we're talking about. And so we looked into the Sierra Nevadas where a lot of these mines take place. The gold you can see is all of the gold mines, and yeah, it's a big cluster in the Sierra Nevadas. So using the GBIF data, we plotted all of the observations before 1920 in the northern Sierra Nevada foothills on R. And we looked in this historical data because the Sierras were interesting to us because they were isolated from the Spanish who had previously visited California, but they hadn't necessarily made it into the Sierra Nevada foothills. So it would be a good location for us to consider without any effects of colonization. But now we just have a big map of all of these observations. So what do we do with it? You have to look into it and try to find if there's any interesting patterns or anything that could lead you to further questions. So I looked and I saw that there were some observations that were within these lakes. And they weren't observations of species that you would expect to be found within lakes like a fish. It was insects and it was plants. And the reason for this is because these lakes were actually mostly man-made in the Northern Sierras. So this right here is the Folsom Dam, which was constructed in 1955. And it's just one of the many water projects in the Sierra Nevada foothills that were built to provide water and hydroelectric power to expanding urban centers of California around this time. So that got me thinking about what happened to the species on this land that was flooded. And more specifically, how were their distributions affected? So now we get into a little bit of data collection and data processing. First, we began by collecting all of the data that was from GBIF that is inside the shape of the Northern Sierra Nevada foothills and downloaded that and used the shapefile with GIS. Then to further make sense of the data, we had to subset it to get the occurrence data, the observations from GBIF that are within these reservoir shapefiles, the reservoir polygons. And then finally, we noted that there was a variable of coordinate uncertainty to these observations from GBIF. So just a range in which the observations may actually be from. And so since a lot of the lakes we were looking at, like Folsom Lake, for example, is 400 meters wide, we decided that a reasonable limitation would be 100 meters so that we could reasonably assume that the observations were actually in this land that was flooded. So now that we have the data where we want it, we can go on to analyzing it. So first we have plotted the most observed species that were within these reservoir polygons in the subset by count. So we first started by taking a look at the most observed, which was Allium peninsulae. And Allium peninsulae is a North American wild onion that was observed actually within multiple reservoir polygons. So here we have it plotted before 1965, which was just a year that we chose as a delineation because most of the reservoirs were built around this time. So here we have in black is their old observations, and then in purple we can see all of the observations after the reservoirs were constructed. And then we zoom in a little more to get some more insight from these maps. And just some preliminary observations are that the old observations tend to be clustered together a little more, and the new observations also tend to be clustered together, but we don't see so much overlap between the two. Next, we moved on to some other interesting species like this Vespericola hapless, which is a snail. And the interesting thing about this is that it is considered critically imperiled. It is conservation rank G1, which is just before being considered possibly extinct. And a known barrier to the species distribution is permanent bodies of water that are greater than 30 meters wide, which is a big red flag when we're talking about reservoirs. So we see here the old observations of this species, and it's just 3 observations, all from the same location in 1946 on what was called Mormon Island, which wasn't actually an island, it was just surrounded by some rivers, but it's now under Folsom Lake. So now let's wonder where it is now. It's actually nowhere. It hasn't been observed any other time since then. So this is a very rare snail, and it really speaks to the possibility for the flooding of this land to potentially endanger these very rare and critically imperiled species. So another species that we looked into is called Eremophila alpestris, and it is this horned lark that is found in the northern hemisphere. It's a common bird, but in the past half century, its population has seen a steep decline. So we plotted this bird. Also, again, we have in the black is the old observations and purple is the new observations. And looking at this, we see that the black is far below the other ones, so maybe it moved north. But actually, if we add a little bit more detail to this map, like for example, with these topographical contour lines, we can understand that perhaps elevation played a role in the species distribution shifts. So we see here that the old observation is seen at a much lower elevation than the newer observations have been. So we questioned if there was potentially an elevation component to the species distribution shifts. The way we wanted to investigate this was using an elevation raster data set. So I want to explain a little bit about what a raster data set looks like. It's essentially completely gridded or pixelated, where each box contains the elevation for that area. And so if we have an occurrence that overlaps, then we can extract the data and see which elevation the occurrence was found at. So going back to the Eremophila alpestris, we actually found that the mean elevation before reservoir construction is in fact higher than the mean elevation after reservoir construction, but we do see a lot of outliers. So some future analysis with additional statistical tests would be beneficial in understanding the statistical significance of these findings about elevation. But we wanted to further consider elevation with some other species. So let's go back to the Allium peninsulae, our wild onion found here. And if we look at the box plots for these, also for the elevation, we see that there was an increase in mean elevation from 311 meters before 1965 to 359 after 1965. So we can conclude that there could potentially be an influence of reservoirs on the species distribution, but it is definitely necessary to consider the species ecology, and that could be very interesting to look into, like considering how the birds were affected versus the plants were affected. And then also we are aware that climate change can cause species distributions to shift to higher elevations, so we could consider the effects of that as well as the reservoirs. So some future directions for how we could try to answer these questions. We could begin with a comparative analysis in which we compare the distribution shifts in similar species that either had or didn't have the influence of flooding by reservoirs. We could also use modeling projections in which we use species distribution models to fit them to present conditions and project backwards to where we expect the species to have been and compare that to what we observed with the historical data. And then another way is through centroid mapping, in which we could calculate abundance-weighted centroids of occurrences, both before and after the construction of reservoirs. And we could include this centroid as a component of a comparative analysis as well. So there's a lot of questions that were raised by just my kind of general exploratory investigation into the influence of reservoirs on species distributions. So I want to thank my mentors, Dr. Nat Low and Dr. Avery Hill, for all their help. I want to thank the SSI leaders who were so great, and everyone here at the Academy has been just so helpful, and all of my fellow interns. So thank you all so much, and now for questions.
All right, does anyone have any questions for Audrey?
Thank you, Audrey. This is really great. I was wondering if you had, like, obviously, the older we go back in time, the fewer observations we have to work off, probably. Did you have any more recently constructed reservoirs that you could look at if you had a more comprehensive historical database of observations to work on?
Oh, that would definitely be interesting to look at newer reservoirs, because the age of the observations was a limitation, because like when I originally wanted to look at mining, we can't really consider before and after mining because we just simply don't have the data from then. But yeah, that's a great future direction to consider current reservoirs using iNaturalist data.
Yeah, Audrey, a good place to maybe think about that is in Marin County, because those are newer reservoirs, and the Academy or botanists especially did big surveys of Marin County.
Oh wow, that's awesome.
They're not necessarily the location data isn't always perfect. It might say like a big area, but it might be a place to start.
Right. Great.
Maddie asked her question. My question is, like of the things that you identified, there was like 10 or so. What was the one that was most surprising and why?
I think I was really surprised by that snail, the Vespericola. That was crazy. And we considered if potentially like filtering out the coordinate uncertainty may have hidden some observations, but actually when we looked at the unfiltered data set, there still were no observations of that snail ever. It's very rare. So that was, yeah, kind of a crazy find that it was inland. So it really makes you consider like if it could have looked potentially different if its environment hadn't been completely destroyed or changed.
Speaking of the snail, you mentioned like it's basically almost considered extinct. Is there any talk of people going into the field to where it's once been found to see if they can find the snail anywhere, or is that not really?
I haven't heard of anything like that. There's really like no iNaturalist observations of it, even like just all over, not just within the foothills. So it's really rare. So maybe if anyone's feeling inspired, you can go out to Folsom Lake, Mormon Island, check it out.
You did amazing. That was a great presentation.
Thank you, Katie.
But I guess my question is, out of all the species that you picked, like allium and stuff to look at, why those? Did you have like any like? It could be random. That's okay too.
Oh yeah. Well, like we had just plotted or charted out what were the most observed species, so that we could be using the most reliable data, that it was most reliably within the reservoirs because it had the highest count. And then the species that I chose to show just had the most interesting graphs or maps, but there were so many maps, I could show you all of them. All of those species have all their distribution maps, yeah.
Awesome. Well, awesome job, Audrey. One of the cool things about the work that you did and combining it with community science, this is my department, I'm not just going on and on, but, is that we could launch community science campaigns to look for those species.
Right.
And so because we have this data that you found because of your curiosity, like we have the opportunity to launch potentially campaigns to look for a lost snail or like where else is this onion found.
Yeah.
So there's a cool
that's a great part about like data science being iterative in that way, and then also combining that with community science, which has the power to continue to go out and collect that data for us. That's great.
All right. Well, thanks, Audrey.
Thank you.
All right. Next, we have Nia Gladden, who's joining us from the Georgia Institute of Technology. Was on the other side, and was advised this summer, and actually is also a Georgia Institute conservation scholar. So we were able to host her here at the Academy with the SSI program, but she has been part of the Georgia Institute Conservation Scholar Program for 2 years now. And we were lucky to have her. She was mentored this summer by Laurel Allen.
Good afternoon, everyone. Thank you, Lauren, for that introduction. My name is Nia Gladden. I'm a fourth-year biology major from Georgia Tech, working as a science communication intern here at Cal Academy with Laurel Allen. And today I'll be talking about my summer research project titled Coming Home, where I will evaluate the impact of individuals' connection to places as a motivator of conservation behaviors. So first, I would like to start off by asking, how many of you have ever seen or heard about this species? Yeah, a good amount. How about this one? Yeah. And this one? Yeah. The answer is probably yes, even though you've likely never seen them for yourselves in the wild. These species are incredible looking, iconic species that are widely used in conservation campaigns. But then I ask you, how many of all have ever seen or heard about this species? A lot less of you. How about this one? Even fewer. And this one? Oh, a couple more, but not as many as the other ones. So this is the Miriam's kangaroo rat, the Savannah sparrow, and the Santa Cruz long-toed salamander. But unless you're an expert like many of you are, you're probably less familiar with these species than with the giant panda, even though these are all endangered or threatened species found right here in California. So all of these species need protection, but they are not treated equally. The macaw, elephant, and panda are charismatic megafauna known as flagship species. Flagship species can be defined as a species used as the focus of a broader conservation marketing campaign based on its possession of one or more traits that appeal to the target audience. And none of the flagship species shown here are native to California, let alone the United States. Currently, these kinds of flagship species are what we typically see when we're shown a conservation campaign. Focusing conservation messaging on charismatic megafauna can lead to the public overvaluing individual charismatic species and undervaluing biodiversity itself. This can be harmful when it comes to protecting less charismatic species that aren't able to generate funding on appearance alone. So I wondered, who's gonna support these little guys, and how can we get people interested in the less charismatic species that are also critical for biodiversity? Walking around the museum, experiencing the State of Nature California exhibit, as well as the rainforest and Philippine coral reefs and others, I started to think about how much more of an emotional connection I felt to the California species, simply because I've lived in the same place as these species and developed a connection to California over the past 2 months. Then I started to think more broadly about how powerful our ideas of connection and connection to home are, and began researching sense of place. And the more I read, the more interested I became in sense of place as a tool for conservation messaging. Rahala et al. Defines sense of place as the wide range of connections between people and places that develops based on the place meanings and attachment a person has for a particular setting. And what's really interesting is that they also found that a shared sense of place may enhance human capacity to adapt and respond to change by motivating conservation and pro-environmental behaviors that promote long-term stewardship and transitions towards sustainability, which led me to believe that sense of place could be a really powerful tool in conservation messaging. So my hypothesis was people will be more willing to engage in conservation behaviors on behalf of species from or native to the same place that they are. So I sought to investigate how one's sense of connectedness to a place, so where a person perceives themselves as being from, which could be the same or different from where they actually live, may influence their willingness to engage in behaviors that protect species also from that location, regardless of their charisma. I was interested in two primary questions. First, are people more likely to show a preference for species from their home region over another species? And second, are people more willing to actually engage in conservation behaviors for species that are from their home region? To test this, I built a web experience called 5usbirds.com. If anyone hasn't filled out the survey, I encourage you to do so after this. So I designed the web experience to uncover answers to these two questions, and in designing it, I kept the following criteria in mind when choosing the test species. I wanted the test species to be appealing but not charismatic enough to be considered a traditional flagship species, and I wanted the species to have a similar level of appeal and not overshadow each other in terms of charisma and overall appearance. And finally, there needed to be at least one test species from each major region of the United States to capture a more personalized sense of place for a wide range of audiences. Ultimately, I decided that 5 brown, relatively plain-looking sparrows best satisfied these criteria. I decided on the American tree sparrow, the clay-colored sparrow, cassin sparrow, field sparrow, and sagebrush sparrow. And with the help with Ornithology Senior Collection Manager Mo Flannery and the Curator of Ornithology and Mammalogy, Dr. Jack Dumbacher, I chose these 5 species. So here's a map showing the 5 major regions and the birds assigned to each. So in the northeast, we have the American tree sparrow. The southeast has the field sparrow. The Midwest has a clay-colored sparrow. The Southwest has the cassin sparrow, and the sagebrush sparrow is in the West. So my web experience starts by asking participants where they consider themselves to be from to get them thinking about their sense of home. And then participants are asked to select 2 birds that they're interested in learning about based solely on what is shown here, which is their regions underneath each bird. Participants are then directed to the information page about the first bird that they selected, where they read a basic description of the species, information about their diet, habitat, and conservation status. And then this is immediately followed by the survey questions. I first start by asking, what state are you from and what city or town are you from? And then to test their willingness to conserve, I ask, how likely are you to want to learn more about this species? How likely are you to vote for legislation that protects this species? How likely are you to donate to the conservation or protection of this species? And how likely are you to write to policymakers to protect this species? And finally, how likely are you to support efforts to create protected areas for this species in your hometown? And once the survey is complete, they repeat this whole process for the second bird that they selected. Before I get into my analysis, I wanted to say that I had about 173 total responses after sending out the survey via social media, the Cal Academy social media. I had flyers that I put up kind of around San Francisco, and I did some sampling on the public floor. But unfortunately, I can only use 100 of that because not everyone completed it correctly. But out of those 100, I was still able to get some really cool results. So when I was doing my analysis, I asked what percentage of people selected their home bird first? And second, are those people more willing to take action to protect their home bird versus their second selection? So to answer the first question, I looked at the percentage of people who selected their home bird first, their home bird second, and those who did not select their home bird at all. So those who selected their home bird first is in the darker green color. Those who selected their home bird second is in like the grayish light purple color. And those who selected their home bird or did not select their home bird at all is in the lighter green. Some insights I found are that a majority of the respondents, 57%, selected their home bird. And out of those 57%, 68% chose it first. The respondents who did not select their home bird at all was still a pretty big significant group with 43% of respondents. And then to answer the second question, I looked at each survey question individually, and for those who selected their home bird first, shown in green, and found the average willingness of respondents to conduct conservation behaviors on behalf of their selected species, where 0 is not at all and 5 is very likely. We can see that in 4 or 5 categories, people were more willing to engage in conservation behaviors for their home bird. And in the 5th category, where it's learn more, it was a tie. In the support for protected areas category, willingness was 14.5% higher for the home bird than the second selection, and we'll talk more about that later. So then when I looked at those who selected their home bird second, also shown in green, what we found was that people were more willing to engage in conservation behaviors for their home bird in only 2 of 5 categories. And overall willingness was significantly lower in the donate and write categories, where in the donate categories, it was 32% less, and in the write categories, it was 31% less when you compare it to those who chose their home bird first. And actually in all of them, donate and write were the lowest categories, I guess, because those require a lot more of participants than simply like voting or just learning more. And then finally, I looked at those who did not select their home bird at all, and overall willingness is lower than home bird first in all 5 categories and lower than home bird second in 3 categories. The 2 in which is higher is donate and write. So my findings provide support for Rahal et al.'s hypothesis that connection to place may increase people's capacity to care and take action. In fact, we saw that for this survey question, how likely are you to support efforts to create protected areas for this species in your hometown, where respondents are specifically asked about conservation efforts in their homes. Willingness was 14.5% higher for those who selected their home bird first than the second selection. There appears to be two distinct groups of people, those for whom connection to place doesn't matter and those for whom it does. And in my data, this was 57% of respondents. This 57% is significant enough group to indicate that in support of my hypothesis, a person's sense of place can play a critical role in how we connect with and conserve species, and it can be an effective tool for conservation messaging for more than half of the public to better support and connect with local biodiversity. I plan to continue collecting data from this project and further analyze the current emerging trends. Specifically, I would like to dig deeper into whether connection of place increases or simply correlates to an increased willingness to take action. I also want to look at the role of age, gender identity, and region and how people connect with and engage in these conservation behaviors. And finally, I would like to refine the web design to make it easier for participants to complete both surveys, as 42% of respondents did not complete them both. Further, I would like to conduct more intensive study on the role and importance of place, as it could have major implications for the future of conservation messaging. My data indicates that if we can incorporate a sense of place into these messages, we have the potential to boost place-based conservation and support global biodiversity in ways that would resonate with a large percentage of the public. Place-based conservation, as defined by the NOAA, focuses on methods and issues specific to a particular region, engaging communities in local conservation efforts, leading to more support for biodiversity. This approach can lead to more effective and sustainable conservation practices by fostering stronger connections between people and their local environments. And that is all. Thank you all for listening, and a special thank you to Laurel Allen, my mentor, the program directors, Dr. Lauren Esposito, Dr. Rebecca Johnson, and Dr. Ed Myers, Mo Flannery for helping, as well as Dr. Jack Dumbacher, the DE team, Luigi, Sofia, my boyfriend Jaquez, and the whole SSI 2024 cohort. Thank you.
Awesome. Does anyone have any questions for Nia?
I'm not sure if this is a question or like a statement more so, but I wonder if it'd be interesting you talked about how like the first question on the website is to put in where you're from. And I'm thinking, like, at least for me, like where I go to school, where I've lived for the past 4 years, is very different from the East Coast where I'm from originally. I'd be curious to, in the future, delve deeper into see if there's a difference there of like, if someone's lived in the same area for their whole life, I wonder if someone's lived somewhere for their whole life, if the conservation messaging that you're talking about would be more impactful than if they've moved around a ton, they might have a different kind of sense of place. I think that'd be potentially interesting to talk about in the future. I don't know.
Yeah, I agree. Me and Laura were kind of thinking about looking at how age would affect that, because, you know, younger people are more likely to like be moving around for like college and stuff, as opposed to like older people probably have a more established place. So we wanted to look at that more, but just didn't have time. Finished collecting data on Monday, so it was kind of crunching for it. But yeah, I definitely would like to look at that.
That was super cool. And it's a really interesting question. I was just wondering, like, what do you think about like whether different ways of expressing place could have an impact? When you're talking about like, you know, I think with the birds, it was just like the name of regions. And sometimes like people don't agree on which region the state is in. So I'm wondering like maybe just like maybe like a range map or like something. And I'm wondering if that might evoke something different, or like listing states might like tap into a different sense of place. Yeah, just one thought.
Yeah, that's something I definitely wanted to look at. I know not everyone kind of puts themselves into regions. States is definitely more small, but it would be kind of hard to have like a bird for each one or like a species because birds fly. So they like, they can go anywhere. But I did notice like people from like Texas, they might have considered themselves Southeast instead of Southwest. It was like really weird. Or like people from Ohio might consider themselves Northeast instead of Midwest. So it's like really hard for people to know.
Do you think that age has a factor in how you view conservation? Because when I was younger, I didn't care less about birds. Now I even go out of my way to buy food to feed them. So I think, I think an age group will be a big factor in how people think about biodiversity or taking care of the birds.
I agree. I definitely want to keep looking into the data before SOCNAS because I feel like that's a big part of the picture that I didn't get a chance to look at yet.
I think this is super cool. I think that if you could put your head together with a data scientist, like they could actually build you a tool that would like show you like specific birds based on where you are at that moment. And so you wouldn't have to guess on the region. Like you could like have a randomized way of looking at like a local bird versus non-local birds. You could also ask people like, have you seen this bird? Like you could, like it would change, it'd be a different study, but you could kind of think about, like you were saying, how you were connected to the birds that you knew a little or the animals you knew more in state of nature. So you could explicitly ask people too a little bit. This is super, it was awesome. Oh. All right, so let's thank Nia and then we are gonna take a 5-minute break.
we're nearing the very end. Don't cry. Okay, next up we have Matthew McDonald from the College of Charleston, who was mentored by Ed Myers this summer, who we all admire. Come on up.
Hey, y'all. What's up? I'm Matthew, and I was mentored by the magnanimous Ed Myers. And we focused my research this summer on how the geologic history and geography of the Baja Peninsula in Mexico affected the Baja Coachwhip snake. And so what we were assessing was genetic divergence, which is very common across nature and occurs in the allopatric method when a once connected population then becomes separated, often through a geologic event, such as formation of a mountain. And as we'll see in a second, also the formation of seaways. But a more local example, I'll throw out California actually, is the Ensatina genus of salamanders. It forms a complex, but they're, depending on your definition, all one species that diverged because of physical isolation. You can see in the Central Valley, they really don't occur, but they do occur in the north and south coastal ranges, as well as the Sierra Nevada mountains. So this concept is very common across nature, as I said, and again, it's common across the Baja Peninsula in Mexico. And so it's common even across birds. If you didn't know birds, many birds can fly, in fact. But even this, even with birds, their gene flow is limited across large areas. One hypothesis for genetic divergence of populations of animals across the Baja Peninsula is 3 seaways, which formed at different times. This one being the oldest, which a seaway is simply land being flooded by water and therefore not being connected. This pattern is also reflected in reptiles, some lizards, as well as snakes like Crotalus ruber, the red diamond rattlesnake, which displays divergence much more recently, slightly south. And then even this genetic divergence is such a common phenomenon. It even occurs in plants like this lovely cactus here, the Sinita cactus. And that occurs along the La Paz Seaway. If I know some of y'all know Spanish, sorry. But the seaway there also causes genetic divergence. So we wanted to assess whether this genetic divergence also occurred in the Mastocophis genus of snakes, specifically Mastocophis phylogenosus, which is the Baja Coachwhip. The Mastocophis genus is commonly known as the whip snakes. They're very fast and they don't like being picked up. They allegedly get their name from their tail, which kind of loosely resembles a braided whip that a stagecoach driver would use. That's what I read in natural history books. There's several different species. Mastocophis flagellum is closely related, and that they're highly phenotypically variable. And that one's eating a rabbit, which is just sick. So I put that in there. You'll note the whip snake genus is quite widespread and appears to be affected locally by geography. So you'll notice the Mississippi River Valley has 2 separate populations for the most part that occur on the east and west of them. And this geographic pattern is mimicked in parts of northern Mexico and then to the south there. And that's due to some arid foothills. Although previous work, genetic work, obviously is suggesting that this is highly influenced, or it's a genetically diverse complex. There's been little work done in the Baja Peninsula. So if we zoom in, obviously, there's been a lot of work done in Arizona, Southern California. But for the 775-mile long peninsula, there's only one genetic sample. Right? That's longer than the coast of California. So there's a lot of cryptic diversity, genetic diversity that may be missing there. And mind you, this is a whole other species, the Mastocophis flagellum, but it does have interesting genetic relationships with the flagellum. So that leads us to our driving questions. We wanted to assess, as I said, the geologic history of the Baja Peninsula and whether it strongly is concordant with the phylogeographic patterns of the Baja Coachwhip snake, that is the formation of distinct genetic populations. And then a closely related question, is this pattern also replicated in other taxa and other animals? So my work was primarily data processing and visualization. Unfortunately, I was not able to go out and catch the snakes. I would have loved to, but there was many people, including Ed Myers, that did that. And then all of the genetic work that laid the groundwork for what I did, I believe was done by Dr. Michael Ewan. So thank you for that, and I'll get into that now. So my research focused on low coverage whole genome sequencing, as opposed to what many other genetic studies do, is focus on one gene or maybe even several genes. Mine takes the entire mitochondrial genome of the snake, which is 13 genes, and estimates their base pair alignment only a few times, on an average of 5 times, variable from 1 to 10. And what this allows us to do is rapidly and fairly accurately, due to just the raw sample size, estimate the total base pair placement of the species compared to individuals, and therefore uncover its evolutionary history more holistically. So after we do this, or not after Michael did low coverage whole genome sequencing, we have tissue samples that were collected from both live specimens and ones that were roadkill. That's pretty common to find snakes roadkill, unfortunately. Tissue samples were taken, then genome sequencing occurred. Then we had to separate out mitochondrial DNA from nuclear DNA. And that was done with a process that even works in my old Mac, in the Mac terminal, called MitoBIM, and is described very vaguely by this graphic here, in which you take all of the mix of nuclear DNA and mitochondrial DNA and use the program. You can separate out just mitochondrial DNA. From there, then, as many of the fellow interns know, you can generate an alignment through the Genius software. So what you see here is every single row is representative of a different specimen. So 73 specimens were collected in total. I believe in this analysis, there were 68 that we had time to run reads on. But you can see their base pair alignments are right there. And generally, they're highly conserved because it is the same species. But in highlights, you can see where possible differences occur. And then we had to visualize this data through generating phylogenies or genetic trees. Below, you'll see a very rudimentary example, also generated on my very old Mac terminal, that just shows the different file names for different specimens and how they're related to each other. And then there was also the process of using the Beast software, which was a little more complex. And then once we generated these genetic trees, we had to visualize them, compare them against where the species are actually occurring. So then we can get a grasp on what populations look like genetically in comparison to each other. So we use things like RStudio, Simple Mapper, and GBIF. So here's one of my phylogenies here. It's a maximum likelihood phylogeny for both Massacovus phylogenosus, again, the Baja Coachwhip, and Flagellum, which is the, it depends on what you want to call it, but we collapsed it into the Flagellum complex. It has some pretty interesting results with some pretty high statistical support. That you'll notice that the green there, that's Massacovus flagellum, is most closely related to the Massacovus phylogenosus. It's not, the pink clade here is not closest related to other members of its species. That doesn't make any sense. And similarly, this is, there's also an interesting pattern going on here where the topmost clade there, the EAM stands for Edward Myers, he collected those species. the Phylogeognosis species in the Cape region of the Baja there, is not closely related to the pink clade, which is on an island. It's an island population. Even though they are closely, that island's only 15 miles offshore or so and could represent a possible colonization event in the past. So this is a Bayesian inference and also has high statistical support for these large clades, and is showing the same pattern of one species being closely related to another and excluding the other clades. So what we encountered here was a pretty common problem. It appears this is likely due to incomplete lineage sorting, which occurs when populations are diverging. Some genes are simply conserved randomly. So when you are observing them, it gives the false impression that they are more closely related to each other than they are in actuality. And so that's why it's important to use low coverage whole genome sequencing. We can go back through further analysis and uncover more holistically their evolutionary story, and as opposed to just one or two genes. This figure is illustrating that in marsupials, which is also a very phylogenetically complex. If you follow something like this green tree here, it appears that one clade will be much more similar, will be highly evolutionarily distant from the one here, say. But if you follow a different evolutionary lineage, say the black line, you'll see that they're quite closely related. Again, underlining the importance of looking at the whole genome. So nonetheless, while these results are very strange, there was a pretty high correspondence between one of our hypotheses and the results that we found. The southernmost clade here in light blue occurred just after the La Paz line, that hypothesized seaway that occurred approximately 3 million years ago on the Baja Peninsula, with likely some zone of hybridization. That's why you see the orange overlap there. Due to, I mean, obviously it's over land now, so it's possible hybridization is occurring. As far as the rest of the peninsula goes, obviously it doesn't appear like there are there's much population structure, but this also could be to limited sampling. 73 snakes is a lot of snakes to catch or find dead. But nonetheless, on a 775-mile peninsula, you're going to need a lot of samples to really tease out what's going on genetically. So as far as next steps go, of course, the common refrain is we need more data. So of course, we'll throw that one in there. But to tease out incomplete lineage sorting, we would need to compare it with nuclear DNA, which we also have from our tissue samples, but we have not looked into yet. And then also we can look at how more local conditions, that is environmental conditions, are affecting or driving genetic divergence, and not so much these large macro scale geologic occurrences such as mountain formation. And that would be measuring environmental genetic isolation through environmental distance, which is not necessarily physical distance, but the degree of difference between two environments, which can be, even when they're quite close to each other, can be quite different. And that can also drive genetic divergence. And also returning to this island, I would love to go to this island and look at more species and determine if via some simulation events, whether a colonization event occurred, and why that pink clade that you saw was so closely related to flagellum and not its other phylogenosis conspecifics. So to recap, little population structure was found throughout the majority of the peninsula. However, in La Paz, there seemed to be population structure. And other than this cactus that I had previously mentioned, the Sonita cactus, the genetic divergence occurring both north and south of the La Paz line, of that former seaway, seems to occur in many other species. So the beloved Miriam's kangaroo rat that some of us had mentioned earlier also displays this to some extent across various seaways. If the Coachwhip and that little fella came across each other, I don't think it would end well, but nonetheless, they co-occur to some extent. And this also occurs in other snakes as well. So there's also another highly divergent, wide-ranging species complex known as commonly just as like night snakes. It's Hypsoglanus sleveni is the specific species that is having divergence in the north and the south of the little pause line there. So for acknowledgements, there was a lot of people that helped me. I apologize that I didn't put I didn't put your proper title there, but Dr. Michael Ewan did all the sequencing, and several different collaborators that are working in Baja that I unfortunately did not have the pleasure to meet. And who else on there? Dr. Ed Myers, Dr. Lauren Esposito, Dr. Rebecca Johnson, many different folks. Shout out to my parents and my girlfriend as well. Wait, what? Oh, I had one other slide, but it was just a picture of me with a snake that said question. So thank you.
Does anyone have any questions for Matthew?
Where did you get that picture of Ed?
That's literally a screenshot from his Gmail.
Oh, okay.
And I blew it up because I knew it had bad resolution.
Soon to be replaced with the paper plate drawing.
Even better, yeah.
Any questions?
Do you know anything about like the weather patterns in the south of Baja that might have driven like a colonization event?
Absolutely. Yeah, that was something I'm looking into it for further steps. The Baja region actually receives a significant amount more of precipitation, as much as twice the amount of precipitation at times, like say like the month of October. That's a few more millimeters of rain on average. There's some areas that are so different than the largely arid interior of Baja that it's actually some carnivorous forests. So it's quite different ecologically, and I'd like to examine if that's causing some divergence as well.
Thanks.
Yeah.
Hi, Matthew. That was a great
Hello.
talk. That was really amazing.
Thank you.
I have a question. I came a little bit late, so you could have answered it already. But how did you know that what you were looking at was so divergent that you had to do meta sequencing instead of just looking at one part of the genome?
Are you talking about the difference between Mascophis flagellum and Phylogenosis?
Yeah, I think so.
Yeah. So Mascophis flagellum doesn't occur on the Baja Peninsula, and that's reinforced not so much morphologically because they look very similar in both the flagellum species and the Baja species are very, very variable. So you can't really tell them apart by that, but you can tell them apart by range, and that's reinforced genetically. They are divergent from each other. So when that don't trust my phylogenies, that I think due to incomplete lineage sorting, that's giving a false signal that these were not properly speciated. Yeah, thank you.
Hi, Matthew.
Hello.
I found your talk extremely informative, especially learning that birds can fly.
Oh, thank you.
Thank you. Yeah, I was wondering on that line, what are the general dispersal abilities of these snakes? I'm assuming they might be able to fly, but in general, how well do they actually disperse? Like, have people done mark recapture on anything like these Coachwhip things?
That's a great question. So as far as flying goes in snakes, to my knowledge, no, but there is a gliding snake that can glide from tree to tree just by flattening itself. But more seriously, as far as dispersal goes, I'm not sure. Considering they're quite large and quite fast, I would say their vigility, like their ability to disperse, is quite high compared to something like the Hypsoglanus snakes. I don't know of any market recapture studies though. That's very interesting though.
All right. Thanks, Matthew.
Thank you.
All right. Our last speaker of the day is Daniel Martinez, local here from San Francisco State. And this summer, Daniel worked with Jacob Garneau and Lauren Esposito. Come on up, Daniel.
All right. Good afternoon, everyone. I'm Daniel Martinez. My mentors are Jacob and Lauren, and I have been working with them all summer. Please bear with me. I've just came back from a long trip, and my mind is not at the right state, but but yeah. All right. So getting into wildfires. Wildfires are a natural part of California's ecosystem. Recently, however, there have been an excessive amount of wildfires in the state. Climate change plays a big factor in this, but also people who advocate that all wildfires are bad. Wildfires need to happen in order to get rid of extra shrubs, trees, weeds, etc., which can be a fire hazard if there's too much of it. People have done research in the past for vertebrates to see how they react to wildfires, how they adapt, and how they come back. But we seem to know very little about how arthropods come back to recently burned areas, or if they even leave in the first place. So our goal is to determine how leaf litter arthropods return to a habitat that has recently endured a wildfire. To achieve this, we must take samples of leaf litter found in different fire areas that occurred in different time periods and locations. We must also collect leaf litter from unburned sites to compare them to the burnt ones. So I went to 3 different fire locations in this trip. The Slater Fire in Klamath National Forest that occurred in 2020. The Frying Pan Fire also in Klamath National Forest that occurred in 2014. And the Megrum Fire in Six Rivers National Forest, which occurred in 1999. I went with Jacob and Procrit, someone else from Lauren's lab. And the trip was from July 15th to July 23rd. We sampled for 6 days, 2 days for each fire site, and 10 burnt and 4 unburnt locations for each fire. These dates are important to see if time periods affect how arthropods recover from wildfires. So each fire was sampled with a 7 by 1 meter quadrats. We tried to find the best spot with good leaf litter, and once we find the spot, we put the meter square on top of it, and we sample everything inside the square. So before we collect the leaf litter, we record data. Some of them include the date and time, latitude and longitude, the elevation, land type, slope and direction of the slope, distance to the 3 nearest trees, the temperature of leaf litter surface, the depth of leaf litter, and soil pH level. So this is the temperature gun, how we take the temperature. That's how we measure the distance to the nearest trees. That's how we measure the depth. And this is how we gather the pH levels. So sifting. We had 2 sifters that me and Procrit used, and we used sifters to gather the leaf litter. Then rather than having something big that'll collect more than what we need. And so I'm gonna show you a demonstration right now of how we did it. So this is the sifter, where it's basically tied to the end, so obviously the leaf litter doesn't fall out. And in the middle, there is a filter, so only like the small things and like the soil will go through. And it is honestly hard work. At first, I was using both hands, but I found out that that's like very exhausting, so I just used my right hand. So I grab it like this, and I start shaking like this for 5 minutes. Obviously, there's nothing in it, but it would fall. It would, fall through, and then we would toss out the large litter every minute. And once we finish, we open this and we pour it into this bag, numbered bag. So this is bag 33. All the bags were they had random numbers. So it was weird, but this would be the bag. So yeah, and then afterwards, that's how that square, that's how it would look like after we collected all the leaf litter. And then our last step was to get the pH levels, which was basically mixing the soil with distilled water and leaving it for 2 to 3 minutes. And then that would be it. We would pack everything and head to the next site, and we would do 7 different sites per day. But it wasn't finished just yet. Once we came back to the cabin, we had to do more work, the Burlese traps. So these traps are used to extract arthropods from compacted leaf litter. How the trap works is that the lamp makes the arthropods that live in the litter want to bury themselves in the dirt since the light can be too hot for them, as well as make the soil dry, which they don't like. Once they bury themselves, they fall into the funnel and into the cup that we place underneath. So the materials that we use to make this trap include buckets, filters, paint sticks, funnels, reptile lamps, and cups. So this is how we would make it. We would put the funnel on top first, and then the filter, and we would place the cup underneath the funnel. Then we would pour the soil or leaf litter onto the funnel, and some excess dirt will fall into the cup. So what we do is just take it out and put it back on top of the leaf litter. Then we fill in the cup with ethanol, label it, and place it back underneath the funnel. Then we put 2 sticks on top of the slides of the bucket so the lamp can rest on it. Then we place the lamp and we turn it on, and we leave the traps for 24 hours. Some bags can require more than 1 Burlese traps. Some can have as little as 2, others as much as 5. And then this was, I think, the first night. This was a row, and then this was in a closet. We did all of that. So the next day, we take the cup out of the bucket. We pour the cup into a Whirlpack plastic bag. The cups that contain the leaf litter collected from the same site go all into one bag, and we pour a little more ethanol if needed. Each plastic bag has a label with information about it, like the date it was collected, what fire it's from, and which leaf litter bag it came from. And we collected a total of 42 bags of these bags. And then we stored the bags in ethanol, in a container of ethanol. So just in case if it leaks or anything, the ethanol won't evaporate. And then we throw the remaining dirt from the Burlese traps away and replace them with new leaf litter collected from the next day, which repeats the process. And that was basically my field work. This is in the lab now. So obviously I just came back, so I haven't examined the leaf litter that we just got, but I have looked through a couple of leaf litter bags that Jacob has collected in the past. My strategy is that I pour it onto a petri dish, and little by little pour it little by little and take out all the arthropods that I can find and put them into one vial that contains 95% ethanol. So these are some of the arthropods I found. Includes spiders, a parasitic wasp that I didn't take a photo of, ants, weevils, I don't think that's a weevil, but weevils, pseudoscorpions, mites, centipedes, thrips, which are those, I didn't know what they were until like now, and many more, many tiny organisms as well. I haven't started lab work yet because my internship is a year long. So I still have lab work to do. I will start that when my school semester starts, and I will be sequencing DNA from all those arthropods, and we will be able to identify these organisms by meta barcoding. So the lessons I've learned. I learned that fieldwork can be extremely exhausting, especially the leaf litter, but it can be very fun at the same time. I also learned that sorting through leaf litter can also be time consuming, but it's exciting to find all the cool arthropods. And never leave ham out of the fridge for too long, or you will end up in the ER. And remember to wash your hands as well when you're doing field work. These are other photos from the trip. I can talk about each one. The scorpion, that was the one and only scorpion we found. It's a scorpion that Jacob is studying, so it was definitely a cool find. We got a leaf with like a cool pattern that I don't know why it has that pattern, but we found a lot of those. Then we got a helicopter with a bucket of water because there were like, I think 2 wildfires happening while we were over there. But we were like at a safe distance, so we were okay. But we saw the helicopters fly by. There were a lot of deer, a lot that came crossing our cars, and so I just decided to take a photo of the deer. And then our last day, we went to the Oregon Caves National Monument and Preserve. We found a giant salamander in there, and that was my first time being in a cave, so that was cool. Also, my first time in Oregon. Here is a longhorn beetle, and it bit me. I didn't know beetles can bite you, but it bit me. Here's Jacob. He got stung by a bee in his ear, and he has a cloth in his ear. He said it was very painful, lasted for many days. And it's so and he also got stung a second time on his finger, but that one wasn't as painful, according to him. And the last picture, long story short, we went into a road, a one-way road to collect leaf litter in those sites. And when we came back, there were 2 cars in front that were blocking our way, so we couldn't get past them. So what we decided to do, we decided to go back to look for more sites. That took us about like 3 hours. We came back and the cars were still there. So we had no way to get out. So what we decided to do is we decided to cut the bushes near the car using Procrit's like machete knife. And we cut like a huge chunk of bushes so our car can like pass through. It was definitely a bumpy trip, but I had fun though. And I would like to thank the Arachnology Lab. I'd like to thank Lauren and Rebecca, even though she's not up here, but I'd like to thank her. And I'd like to thank, of course, the rest of the SSI interns. And yeah, that's basically it. Thank you.
All right. Does anyone have questions for Daniel?
That was a really fun talk. Thanks, man. So I know you're trying to standardize your sampling. So you're sampling at unburned sites and then burned sites. But are there any historical samples that you could use as a baseline that are now burned sites? Does that make sense?
Like burned sites from like a long time ago, or?
Yeah, like somebody's gone out and done leaf litter sampling, let's say like 40 years ago, and that site is now burned.
Not that I know of. I can't really answer that question. Yeah, I'm not 100% sure.
Could you elaborate on what happened with the ham?
I think I got food poisoning from the ham because we left it out from the fridge, and I ate it with a sandwich. It was either that or I was also eating snacks without washing my hands, which is gross. Never do that again. But it most likely was the ham because, yeah.
I'm glad you feel better.
Thank you. I feel well rested. No, not well rested, but I feel my stomach feels better. Yes.
What was your, I guess, favorite part about the trip that you would say?
I would say my favorite part was, I would say the views. There were like a lot of beautiful views in California and in Oregon. And I also like spending time with Jacob and Procrit. They were great. And I also like to thank Jacob for taking me to the hospital. Yeah, he was incredible this trip. Yeah.
Hey, Daniel, I was wondering if you had any favorite music that you listen to while you're out in the field.
That information is not available at the moment. No comment.
All right. Thank you, Daniel. All right. So that concludes the amazing talks of our 2024 SSI cohort. I can speak for both of us. I think when I say we're super proud of all of you, you did amazing jobs. It was so great to not only hear more about the research that you've done, but for me to really think about how you've all developed as researchers and communicators over the summer. Think about like those first Instagram stories you all did the first time you were talking about yourselves and what you care about, and how that, I mean, those were amazing, but you think about like how you've really employed all the things that you've learned this summer to give these great talks today. So just kudos to you. It was really great to get to know you and to hear all of you about your great work today. Thanks again to the audience and for everyone who came and listened to all these amazing talks, the folks online as well. It really is a village of folks who helped shuttle our interns through the academy experience, and it's really nice to see so many folks supporting all of our students.
I think with that, we can just give all the speakers today another round of applause. And for all the mentees, we're going to take a photo. So stick around. And our t-shirts came today.
We also have some snacks.
Yeah, and for everyone in the room, we have a toast and some cupcakes. So if you have time, stick around for a little longer. Thank you.
Thank you. Thanks also to everyone who helped with tech support today and ran the live stream. Felicia, Ali, Guillermo, Alwyn, all the folks that helped out today. AV, thanks so much.